So the goal of this natural history study, and I'll make sure I'm not touching the screen here, is to really advance our understanding of the disease. And like I said, it's progression over time to prepare our community for clinical trials so that we really have, as Wendy has always kind of taught us, to get our toolbox ready so that we have all the necessary tools to be able to move forward with the FDA. It helps us identify meaningful endpoints for measurements in future clinical trials so that we can see kind of where the children's baseline performance is. And if we introduce a therapeutic, do we see any improvements over time based on that therapeutic intervention? The assessments that our children, not all of them did every single assessment, but the assessments that took place were motor function. So they looked at our gait, our children's gait, how they're walking, a PT and OT assessment, neurological exam, cognitive exams. We did brain waves with EEG and blood samples. If you have not participated in our collection of blood samples, I'd encourage you to do so. It's Combined Brain, which is a consortium that we're part of, and Simon Searchlight with one blood draw. Then we're going to be identifying biomarkers in the children's blood, and parents and siblings can contribute also. They're right by the front desk when you walk in, and they will be here all throughout the day today. So our organization and how we have grown, and we've grown a lot. We have a new name. We went from Advancing CTNNB1 Cures and Treatments, merged with Seesaw, and we are now CTNNB1 Connect and Cure. We have a new logo, which you've probably seen on a lot of our marketing materials and website, and fabulous new members that have really helped us move our core initiatives forward. So I'd like to just, in addition to our board members that were on the board before, we've added Effie Parks for strategic partnerships, which I'm sure many of you know who she is. Ashley Swift, who's doing outreach and family onboarding. Tony King, who works on our kind of development and grants. Emily Amerson, who's been doing an amazing job on our social media, and she also works on a lot of our strategic partnerships. And Annie Wood, who was with Seesaw previously, who does a lot of family engagement and runs our Connect and Cure podcast. So some kind of new initiatives over the past, I'd say, 6 months that we've taken on is in addition to Simon Searchlight, where we have our registry. We've created another registry with Citizen, which is really a collection of medical records. You basically sign up that you're allowing them to collect your medical records, and it's an online database of all of the children's medical records and doctor's notes. So it functions as a different type of registry for us. And that's in addition to Simon Searchlight. We've joined the consortium of Combined Brain, which is for other neurological disorders and neurodevelopmental disorders. A lot of them are all members. And we have a biorepository, which is basically a patient-owned blood bank that we're starting with them, and they're collecting blood as well. They're in the front desk, in the front area. You can ask questions. Probably Genetic is a really amazing opportunity for us. They've created a screener for cerebral palsy patients that you can go if you're kind of searching, in that beginning of that medical odyssey that we all went through of trying to figure out all the symptoms you Google about your child, like what could this potentially be? They offer a screener, and if you're flagged basically for CTNNB1, they're offering free genetic testing. And this is really important for us in growing our patient population, which is a main goal of ours. And so it's just another way to hopefully get more patients correctly diagnosed. And then we have our podcast that's run by Annie Wood, the Connect and Cure podcast, just to keep the CTNNB1 community and outside community informed of what's going on. So our goals are really to raise awareness, grow our patient population, support our families, and empower our community. So as we kind of start to think forward about where are we going as an organization and for our disorder, we're kind of gathering I like to call it our preclinical toolbox. How do we set ourselves up so that when we are ready to go with the therapeutic, we have all the necessary, assets to be able to move forward. So we have, our registries with Simon Searchlight and with Citizen now. Now we've done this natural history study with Columbia, which is a really robust data set. We have our biorepositories and animal models, cell models, and we're looking at biomarkers. So, something that Michele and her team have been looking at, and we'll be able to look at more so with the human-derived cell lines that we have, is kind of the variant impact where your child's mutation takes place across a gene and how that affects protein function. We're also looking at genotype phenotype correlations. So what does that mean? It's where your genetic mutation takes place and how your child presents with their symptoms. And if there's a correlation, that's something we're trying to understand. As far as therapeutic development, Michele and her team at Tufts are working on a novel kind of small molecule that's been showing to be very effective, and they'll talk more about that. And also gene, DNA, and RNA-based therapies. And we're looking at these therapies in both animal models and human cell lines. So I just wanted to close with this quote, which is something that when we were starting the organization drove me, and it's, if not us, then who? If not now, then when? I want to thank you so much for bringing your children and helping further our research. It's not only for our children now, but it's for all those future generations of children that are yet to be diagnosed. I think it's really powerful that we're all here together and that we have the power to change the trajectory of this disease for future generations. And that means the world. So thank you very much. With that, I would like to introduce Dr. Wendy Chung from Columbia.
Thanks, Lauren. So really thrilled to be here today. I will say I'm going to go pretty quickly because I've only got about 30 minutes. But all of this is being recorded, so you can come back and hit the replay button as we go through. As Lauren said, I represent Columbia University, actually for 7 more days. And then in a good way, not entirely planned for this reason, but in a good way, the team is going to be moving to Boston Children's Hospital. I'll be in July 1, the chair of pediatrics at Boston Children's and Harvard. And in a good way, I want to message out to the community. This actually ends up being really powerful. In part, you're going to hear from some of my friends who are Boston located, Michele and Jerry and others as well. So it's going to coalesce us to be able to work even more closely together. But also because Simon Searchlight is coming with me, so let me be clear about this. And the Columbia Natural History Study will continue as the Boston Children's Natural History Study and also have partners who are New York based. And so this actually brings more resources to bear. We don't lose anything with the transition, and hopefully anything for you all as participants looks exactly the same. So let me just reassure you of that. But as we do this, hopefully even stronger as we go forward. So within this, Lauren already alluded to Simon Searchlight. And let me just say one little thing about this community in particular. You guys are awesome. And I can say because I'm in the rare disease space and I see lots of different communities, but the fact that you've really united and been able to put together your strength is really a tribute to your leadership and the vision that they've had in doing this. And I just want to say it doesn't happen in all rare disease groups. And I'm going to say it, and I'll say it if you ever like to speak again, stick together within this and stay united as we're doing this, because even though your numbers are growing, they're not big enough, and we want to be able to have that impact as we're going forward. Powering behind this is Simon Searchlight, and I'm committed through Simon Searchlight and what we're going to be doing at Boston Children's. Can you still hear me? Can you hear me? Okay, I'll keep going and I'll try and speak loudly, and I hope those of you at home can hear on the recordings okay. So as we're doing this, and as Lauren accurately said, we're trying to get multimodal data, and we're trying to think forward in terms of what we need for clinical trial readiness. And it's really important for us to know about as many people as we can and know over time how things are changing. So let me just say that I know this takes a lot in terms of this, but it's really important that we do this now. As we're doing this, we realize we have kind of a standard battery of conditions that we use for individuals across neurogenetic conditions, and we feel that we need to fine tune them for each community. So I would call this phase 1 as we're doing this, and the in-person assessments were really important for us to figure out where we need to slide around that sort of scale. We also want to hear feedback from you all. We've been doing it over the last 2 days, but continue to give us feedback in terms of whether or not you think the measures are correctly capturing your children or your loved ones, and we can slide those things, and or in some cases we may need to be able to make up new measures as we're going forward. We are committed in Simon Searchlight to make this an iterative and reciprocal interchange, and so Erica Jones, who's actually here with us today, is part of our communications team who helps us to be able to communicate that information back. Give us feedback whether or not we're hitting the mark as we do this, and as Lauren already said,
As I'll talk about, the blood samples are helpful and freely available to researchers globally. They're de-identified, so no one knows who you are, but we try and, if you will, multiply your investment in terms of the energy that you're putting in. And the same is true for all the Simon Searchlight data. We share it freely availably to any researchers doing this. You're going to hear a little bit later, but one thing we do with some of the cell lines or some of the blood samples is to make cells in a dish, mini brains in the laboratory, things to be able to test out some of the things that you'll hear in terms of preclinical studies. And this helps us test out things without having to endanger your loved one, because we can do it in a dish and there's no concern in terms of toxicity. And believe it or not, we can take cells from the blood and make those into brain cells. They're not perfect brain cells, but they're pretty good close approximations, and even make little mini brains as we're doing this. Important also is that at some point for the community, if you're enrolled in Simon Searchlight, we have an option of what we call research match for any researchers around the world to be able to invite you to studies. You can say yay or nay, you want to do them or not. But importantly, when it comes to a notification, for instance, for clinical trials, this will be one of the important ways that we get the word out and invite people. So just another plug, another reason for doing this. So let me very, very briefly tell you what we have been learning so far from the Searchlight data. It's really amazing that we've had already 222 people from around the world, and this is global for those of you online as well, sign up for doing this, but we also understand that not everyone has gotten through all the steps. And so the numbers shown along the bottom are how many people have gotten through this. And the good news is about 2/3 of you have gotten through the process of being able to give us a copy of your genetic test report, so we know that in fact you're in the right club, and being able to start doing some of the online surveys. But for those of you, and I know several of you have been checking, if you get stuck, just check out here with the Searchlight team, or give us if you go online, there's a place you can email for help to do this. This is mostly for the researchers who may be watching. This is an extensive battery, intentionally so, and we know it's a lot of time and energy, but we do this again so that we can be able to be ready for what researchers are looking for, to understand both what the outcome measures, how we will detect if there's any change, as well as anything that we might need to be particularly careful of. And I point out the annual column on the right. This is not one and done. We need to keep doing this over time as our children are growing up and as things are changing. We also know that there are additional things we need to do to fill in the gaps based on what we've been hearing from researchers, to say, even if you've been participating, there are going to be new things showing up on your dashboard. So online, this is pushing you out to show you what you need to do. Okay, so this is obviously a genetic condition. For the researchers mostly out here, I'm showing what is going to be an opportunity and a challenge. And I apologize about the technical details. But for most of you, and I don't expect you to sort of memorize all of this, but shown in yellow are individual genetic, each person here is represented by a dot. And if there's we call this a lollipop plot. So if the lollipop is taller, then it means that more than one person in the community has the same genetic variant. And although this might feel overwhelming because there's so many different genetic variants, so shown across the bottom is the gene, and each of these is a different position along the gene. So for most of you, you're an N of 1. You are the only person, your child is a unique person out there in the world with their particular variant. So it might feel very lonesome, but it's not, and that's the good news. Okay, now you can hear me even better. So for those of you out there, again, this is the lollipop plot, and each person is represented by a dot here. The ones in yellow are ones that we think result in half of the amount of protein that's produced. And so even though each one of these genetic variants might be either rare or individual, collectively, if you take all of those yellow dots together, we think that it's affecting the brain in the same way. And so in a good way, we're going to use the power of that as we think about strategies moving forward. There are a few people that are a little bit different that are shown either mostly in red, or the ones I would say that are particularly different. And so people in the scientific community are looking at those. For those of you technically inclined, we call them missense variants. And they're looking at those to see if there might be something a little bit different. So I say this because you're going to need this to understand a couple more things that I'm going to say. Within this, as I said, it's at the Simons Foundation, we've created a library of these induced pluripotential stem cells. These are extremely expensive to make, but we've done this on our dollar to make sure that this could power the research community. And again, these are freely available. I put this up here mainly for the researchers so that you can see what we've got in the library. And again, all of these able to be used in terms of any of the studies you might want to do. You simply pay a small shipping fee in terms of being able to get access to this.
Okay, so what have we seen in the community? So number one, this is an age distribution showing along the x-axis how old our folks are who are in Simon's Searchlight. And although it's nice that we have individuals up until the age of 21, I'll point out that we only have individuals up until the age of 21 in the community. That's not to say they're not out there. I truly believe they're out there. We just haven't yet identified who they are. And as you've been hearing from Lauren, they largely haven't been tested. For those of you who are in your communities and maybe thinking about your children getting older, and I call it aging up, do help us think about how to reach that community. I've been working really hard on it. I hope we'll have a story coming out in the Atlantic, actually, telling the stories about this community, because this helps us, in my experience, with knowing what to expect in the future. And it's really critically important to know what to expect in the 30s, 40s, 50s, and 60s so that we can prepare in terms of our children as they're growing up. So just put that out there if you happen to have any ideas about how to get the word out. I'm going to go through this quickly because I don't think it's any surprise. And again, we're going to have this online. This is mostly affecting above the shoulders, not 100%, but mostly in terms of the brain and behavior. It's globally, everyone in the community is affected in some way with a variable severity. We do know that this is affecting especially the body in terms of the movement, but also in terms of, for instance, speech. I'll say hearing and seeing the assessments that we've been doing over the past couple of days, literally with our own eyes, we know that our folks can understand better than they can get it out in terms of being able to communicate. So I'll just put out there that that's one of my hopes in the future with developments of additional assistive devices, that we can do more in terms of expressing ourselves. We do see some behavioral conditions as well, but I'll say that not everyone has those, but issues in terms of autism, anxiety, problems with attention and focusing, all things that we see. And then again, just getting a little bit deeper in terms of some of those issues, all things again related to the brain and behavior, but these are issues that we need to think about in terms of how can we see if we're moving the needle? How can we see if we're getting better in terms of mobility, for instance, in terms of fine motor skills, in terms of processing, understanding, and expression. The good news, and I do think this is good news because of the individuals that we've seen so far, we're not seeing a lot of what I would call medical burden. So not to say that it's not a big deal in terms of what we're seeing in the brain and behavior, but we're not seeing, and again, this is to me good news up until the age of the 20s, we're not seeing, for instance, a lot of seizures or epilepsy. We're not suddenly seeing when the kids are turning to be teenagers that things are, changing radically. So in a good way, that's reassuring to me. From a medical point of view, we're also not seeing a lot of medical burden outside of the gastrointestinal tract. And I do think the GI issues that we're seeing are related to the brain's effect on the gut. So it's all tied together in some way. We're not seeing all of a sudden inflammatory bowel disease. We're not seeing all of a sudden colon cancer, things like that. So even though it feels like, well, there's nothing going on, that's a good thing. And that means we know where to focus in terms of focusing up here. One of the disappointing things to me is that we're also not seeing a sort of magic bullet. So in terms of looking and surveying you about what medications, what therapies, what things you're using, there's nothing that's rising up in terms of, oh my goodness, someone's latched on to the secret in terms of sleep, or the secret in terms of ADHD, or the secret in terms of making the brain blossom with this. We keep our eyes open, and certainly even if it's just one of you who's seen something that you think is truly effective, I'd love for you to directly email me and let me know so we can follow up on it, because you never know where those things are going to come from, but we haven't seen that so far in going forward, hence the need to be able to push forward in terms of the trials and the development of new treatments. So in brief, and again, we're going to post this online, the good news is that we're seeing mostly the brain. I will say vision, and you're going to hear about vision also this afternoon. That's important to me in terms of making sure the brain is capturing things correctly. So the eyes and the ears are windows to the world, and that's important. But in a good way, we're not seeing evolution of things falling apart later on, but they are persisting. The issues are persisting. They're not going away, perhaps not surprisingly. Okay. On to thinking about how we're now, what opportunities we have in terms of treatments. And I do think the good news is that the next decade in particular, there are a lot of folks. One of the other reasons for moving to Boston is there are a lot of folks in the biotech pharma space that are thinking about rare genetic diseases. And so in a good way, part of what I'm going to hope to do is to partner with them in terms of making them aware of many of these rare genetic diseases and what those opportunities are. There are many people working on this. I will say there are many people in what we call universities around the world. There are also many people in biotech pharma developing these things. And what I would say is that there's a lot that's still in the development stage. There's not a lot that in the next year is going to be ready for us. So we do have to plan on this. It's going to take a long-term investment. but there are what I call enabling technologies. There are going to be things that once we figure out how to do certain key things, a bunch of the brain conditions are going to be able to jump on board and all benefit from doing that. So we need to be ready when those things about delivering things to the brain, when we can do it, how we can do it safely, and then be able to pop our gene in or be able to make the adjustments we need to for our gene. But as we have those opportunities, which I do think are coming, we'll be able to hop on board. So just a little bit about this, so that when you're reading the newspaper or you're seeing things come out on podcasts or you're seeing things come out on your Twitter feed, what do you need to pay attention to? I go back to that very complex slide looking at the gene. Our condition is such that, and it's on the right, where we have a variant or a mutation in the gene. The basic problem we have is that instead of producing 100% of the protein, we're producing only 50% of the protein. So we've got two copies of the gene. One's working fine. One basically is, still in bed sleeping. It's not doing its job. It's there. We can see the genetic variant that's there, and we may have ways of capturing that information, but we call this haploinsufficiency. Half of it is insufficient, is not doing its job. And we do think from the number of cells that we've looked at that really is probably the mechanism for all of those yellow dots that we showed before. 50% is not good enough. It's okay, but not good enough. We likely are going to need, I don't know if it's going to be 75%, 80%, but we're going to need to get closer to the 100%. And we're going to need to focus it on the brain, because for all the reasons I've shown you before, the brain is where the action is. That's important and good information, because it suggests that we really do need to focus on the brain and getting it to the brain. We probably don't need to get it to all the parts of the body, but getting to the brain is a very special location in the body. Our body protects our brain. It's a very sort of sacred place. And so getting in there, as I'll show you, takes some tricks to be able to do. As we're doing this, there are a couple ways that we can think about doing this. And I put this up in technical terms, but A, B, and D are what are relevant to our groups. It can either be that we have a gene replacement or gene addition. So we put this back with one copy. Or option D, gene editing, is we fix the copy that's there in some way. So either we use we go in and we literally sort of erase and write in the correct sequence at a DNA level, or more likely in the short term are ways of being able to add back a copy of the gene. Even short of directly working on the gene, and we're going to hear some about this from Dr. Jacobs later, there are also possibilities of using pills. So we'll use the term small molecule, but using other things that you could take by mouth that may affect the brain. They may not be specific in terms of only one mutation. They may work globally for CTNNB1. There may also be opportunities, not just for our gene, but things that will help in terms of memory, attention, other things that will help in general brain function. And so we'll be thinking about those. My prediction is that we'll be at a stage where we're using a pill, we're using a medicine by mouth in a clinical trial before we get to the point of something like gene addition or gene editing. The things like gene replacement, gene addition, gene editing, one of the reasons I say that is the stakes are higher. In terms of doing those, they have the benefit of oftentimes being one and done. They often also have the disadvantage of being one and done. That is, you do it one time and whatever happens, happens. And so from a safety point of view, we want to make sure we're at the point of being very sure that we're safe in doing this. Whereas something like a medication or a pill that we're taking, you can take it, and if it has a side effect or it's not doing what you want it to do, you can always stop it and you go back to being what your body was before. And so, like I said, when I think about it in terms of trying to protect our young people, I think about safety as a very important factor. And so I think we may be going there first. Okay, so in general, and for those of you who remember your biology class from high school, we have our problem at the level of the DNA. It goes through a message that makes, and that's called RNA, and it eventually makes the protein. The level that we're going to be working at for the drugs or small molecules is often at the level of the protein. There are things that you may be hearing about in social media or other things about ASO or allele antisense oligonucleotides. That's often working at the RNA level. And then you may hear about things like gene editing or gene addition, and that's working at the DNA level. So at the end of the day, hopefully it's doing the same thing in terms of making the protein correctly for the body, but it's working in those different ways. At a very high level, gene replacement is the idea that we've got, again, one copy of the gene that hasn't shown up for work, and so we might be able to add back an extra copy of that functional gene. As we're doing that, there are different ways of being able to do that. But for those of you, there might be someone online who's thinking about this. We are not doing it necessarily in all parts of the body. We're not doing genetic engineering to create a superhuman or do anything like that. We're doing this as something that's really targeted at the brain as we're doing this, and we call this somatic gene therapy. So not something that'll be passed on to the next generation, but doing it to try and alleviate the brain problems. I'm not going to go into lots of detail, but one of the big components that we have to solve is just the delivery system. We know what gene we have to give back. How do we actually deliver it? What's the truck that drives it in? If you order something on Amazon, what's literally the truck that's driving up to the house and putting it into the brain? I'm not going to go into a lot of details about that, but we need to get something that can hold our gene, that's safe to do it, that's going to be able to be effective and get throughout all the cells in the brain. That's actually a big factor, is being able to do that and do it evenly. And to say that there are a lot of smart people working on this. The researchers in this community, I don't anticipate are going to solve this problem. They're going to rely on other people to solve that problem more broadly for genetic conditions. And this is one of those things that I said with enabling technologies. Once someone figures out that delivery system, we're going to co-opt that and use it in terms of what we're doing to be able to deliver our gene of interest. To say, if you hear about things doing this, even some of the things you've heard about, for those of you who may have gotten the COVID vaccine and heard about the Moderna technology to do that, believe it or not, some of that same technology is even being used to think about delivery of genes or gene products or things like that. So within this, one of the good things is that, there had to be something good that came out of COVID. So one of the things that was good that came out of COVID was just understanding that technology and being able to develop that further. There are also other technologies using viruses and being able to handicap them so they don't do bad things in terms of giving us infections, but can be useful for delivering those genes. As we do this, there are some things that are a little bit tough about this. We need to be able to do it just right. So we need to be able to give back one copy of the gene. Again, we need to be able to do it safely. We need to make sure that as it goes in, if it goes into our genome, literally into the rest of our genes, that it doesn't disrupt anything else, that it doesn't screw something else up as we're doing it. And so sometimes we think about processes where it doesn't integrate into the rest of the genome, because with other gene therapies, we know that we can cause mischief as we're doing that. There are finally other ways of doing the gene editing and literally fixing the gene. I do want to underscore that one of the challenges for our community about doing that, and remember again that schematic I showed you, is that we have so many different genetic variants, and if we have to fix every single one, and if every single person is different, this does prove a little bit more challenging. There are people that are thinking about how can you do something by fixing blocks of different genetic variants, but I will warn you, as you're hearing about some gene editing strategies, because of the distribution of where our mutations are, this is a little more challenging to me. Not impossible, but a little bit more challenging. I'll also say that you may hear about CRISPR in terms of doing this. The Nobel Prize was given to two incredible women for being able to do this. I'll just plant the seed that some of that early technology has some issues. I won't go into the details for sake of time, but there are some things that happen when you cause what we call double-stranded DNA breaks, which is what the CRISPR technology does. And you can end up creating other problems. And so there are others working on single-stranded DNA, or rather single DNA breaks to be able to avoid some of those problems. And so you may hear about things like prime editing and other technologies that are coming forward. But again, I'll just say that there's some challenges for our community with that. Okay, so let me go back to the idea that I think is better for our community in terms of thinking about adding back the gene. So there are ways of being able to take viruses that are even commonly cold viruses, but being able to handicap them so that they can be that Amazon truck and deliver our cargo of the CTNNB1, so viruses to do that. We can also use this in terms of what we call liposomes. So this is not an infectious agent. It's kind of like that Moderna strategy for the vaccine I was talking about, and being able to coat it in terms of fat. And with that fat, then being able to get that fat to deliver it back into cells. And I do think both of these strategies might long-term present some possibilities. Within all of this, though, and I think this is where we are right now, as Lauren was saying, it really doesn't matter to a certain extent whether it's a pill that we're using, whether it's a liposome, whether it's a virus to do this. We need to get ready in terms of being able to see over time, what is our condition looking like? Is it going to change? What outcome measures are we going to do to see if there's a difference? is there anything in particular we need to be careful about with the toxicity? And only we can do that. So a lot of the other things that I talked about, there are a lot of people that can supply some of those tools, but we need to be what I call clinical trial ready, so that if anyone comes forward and says, I've got a great new technology, what gene should I focus on? If I'm at that meeting, I can say CTNNB1, we're ready for action. We're ready to go. All we need to do is then plug in that into what you're doing. In doing this, as we think about this, there are some challenges, and I want to set expectations as well. There's some challenges in terms of getting to the brain, as I talked about. There are problems in terms of being able to get to all cells in the brain, and the brain works as a system. It works as a network. It works as a circuit. So within doing that, I think some of us want to make sure that we're doing that evenly to get into all of the brain, because if you had a patchwork working with the right side of your head, but not working with the left side of your head, that's not a good thing, right? Because we need the brain working together to be able to do this. In addition, and this is one of the big things, is that we don't know over the life course what window of opportunity we have in terms of treatment. We just don't know. We're not going to know until we start doing this in animal models to start with, to do it safely. And ultimately, we won't know until we do it in people. So I don't want to pre-assume in terms of doing this. I will put out there in terms of hope. Some of you may follow other rare disease groups, and I'll say for Angelman syndrome as an example, a condition that I didn't think that we were necessarily going to have the ability to effectuate change once you got to being 5 or 10 years old or older. I just didn't know what we'd see. I've been impressed that in some cases, at least we can see remarkable changes in individuals who are 7, 8, 9, 10 years old. So I'm not going to say exactly what our window is, but to say that some of the preconceived notions may not be correct. We just don't know the answer until we know. Within this, though, we don't know what we're going to move, how much we're going to move it. And so we're trying to prepare for sensitive measures of change. That is that we want to be able to see the smallest amount of change because we want to iterate. This is going to take this repeated process of seeing something, deciding what changes we need to make, and being able to make adjustments on the fly as quickly as possible. So we don't have to wait 5 years for each step that we do this, right? We want to compress this. Maybe it's 5 months, but we want to compress this and as long as quickly as possible. So as we're doing this, the other thing you may hear about even today are things that we call these ASO treatments. And you may have heard about some of the N-of-1 treatments, either from me or from others in doing this. I don't know whether or not this is going to work, but just to plant the seed, this does offer some of the opportunities that are kind of in between the gene therapy and a pill. And so it's not as easy, but it's not as hard. It's in between. The level at which the ASOs work are at that RNA level. So that's that middle step or the intermediate level. We can manufacture these relatively quickly and efficiently. And the strategy we're going to use is potentially the C portion here. That is in terms of the upregulation. So remember, we've got 2 copies of the gene. If we can make our good copy work twice as hard, work overtime, we may be able to compensate in terms of getting to the total amount of protein or closer to that while using what we have in the body already. As we're doing this, these have been used and can be manufactured very safely and on scale. So I think that's a good thing. I won't get into the details, but some of the researchers who are thinking about this, there are strategies in particular for thinking about genes like our gene, where we need to ramp this up by 50%. And so people know about ways of even making predictions of whether or not we can do it and which ASOs we should use to be able to do that. But in doing this, there are several steps, and Lauren outlined this earlier, but I just want to say very specifically, within this, to set expectations, this takes time. Even if you hear from things today that are very exciting, they're not going to happen tomorrow. And there are a lot of steps that we need to do to get prepared. So we're doing these in parallel. We're doing this on our side with the community. The researchers are doing it on their side with research and development going forward to do things so that we meet in the middle at the same time. That's the trick for us to not have a delay on either side to doing this. In doing this, and the good news, we have the tools in the toolbox that we need. We, at the end of the day, also need to make sure whatever we're doing is safe. So there will be preclinical, before we're in people, toxicity studies. And to me, I'll just tell you, that's a very important gate to make sure that anything we would put in a person is not going to do bad things. We can never predict 100% from what we do in a mouse or a rat to what we do in a person. Those don't always correlate exactly one-to-one, but it will be something that even the agencies, regulatory agencies like the FDA or institutional review boards protecting you will want us to do. I'll also say, and you may hear about this a little bit later today, that once we get into clinical trials, these are no joke. This is actually showing you the schedule of events for a clinical trial that we're doing. And as we're doing this, I want to say that the people who are the first to do this with their families, are incredibly brave and dedicated people. So as you see this, either in our community or other communities, give them a real sort of shout out because they are taking both risks and as they're doing this, do learn from their experience. But just showing you, these are different time points over the course of a clinical trial. Each of these rows is a different thing we ask people to do. Many of your kiddos were with us the last 2 days doing all sorts of going through and getting exhausted with things. Just think about doing this not just once, but doing it time and time again, potentially every 30 to 60 days as we're doing this. Is really dedication. And again, we realize everyone can't do that. So we're understanding, and we're also trying to figure out what are the most important things that we do to make sure that we get that just right. Okay, so rounding out this out and just putting all the pieces together, Siedman Searchlight, a core in terms of doing this, Citizen also helpful in terms of getting medical records to get deeper data, what we're doing with the natural history study at Columbia and Boston Children's, also filling this out and complementing it. And then with combined brain and Searchlight to be able to make those blood samples and biomarkers going forward. As we're doing this, again, I want to reassure people that the data you're contributing is de-identified, so no one knows who you are. It's safe, it's protected, but we need the numbers because each person, each one of those dots on the lollipop, each person is unique in many ways. So let me stop there, and I'm over time, so we'll keep going.
Thank you, Wendy.
So thank you, Lauren and Sean, and the community for the invitation to speak. You'll see on my title slide, hopefully, and also for those online, I posted in the chat a link. I'm trying something because I'm over 10,000 kilometers away from almost all of you to be a little bit interactive. So there's this little QR code here. If you've got a smartphone, you can try and go to my little survey. It's only 3 questions. There's no wrong answers. You won't be easily identifiable. It's all going to be just summarized and hopefully it'll work. And if you don't want to answer the questions, that's fine. So as I said, as Lauren introduced me, I'm from the University of Adelaide, which is in South Australia, which is pretty much the other end of the world. No. Let me see if I can go to the next slide. Why you not want to? Oh, goodness. Sorry, while I have some mouse issues. There we go. So before I go any further, I'd just like to put up this acknowledgement slide. These are the people that have in some way contributed to the studies that I'm going to talk about today. Wendy just spoke about her prospective study with the Siedman Searchlight, which is amazing. And you can see we actually collaborated with Wendy as well on this work. But our study was retrospective. So we really wanted to know what we know. And the person I have put up here as a photograph is Dr. Kayumi. She recently got her PhD and it was part of this work. And so a lot of what I'm going to show you today is actually hers. Any accolades you want to supply should go to Sayaka and perhaps not me. So I wanted to start by just talking about neurodevelopmental disorders. This is how they're typically defined. So they're developmental deficits that produce impairments of personal, social, academic or occupational functioning.
Mark, can you try to share your screen because we still see you and not the presentation?
Oh, why is that? It told me it was sharing. Let me. That's a shame.
Hold on. Hold on a second.
It may have something to do with the recording.
Maybe pause real quick.
Pause the recording. Just give me a solution. Oh, there it is.
Okay, now we can see it. Thank you. Yep.
All right. In the interest of time, I'll probably move ahead. We might take the survey experiment somewhere else. As I was saying, we're interested in neurodevelopmental disorders. Examples of these would include epilepsy, intellectual disability, cerebral palsy, and autism spectrum disorder. I've been working and trying to find genes associated with these disorders for nearly 20 years now. While I'm on this slide, I'd really like to draw your attention to this positive exposure website. Some of you may have seen it already. All of these photos came from there, and I really love the initiative behind that study. I'm just having the greatest of problems with advancing my slides. Oop, okay, so one of those disorders which I mentioned was cerebral palsy, and we came to CP before we started looking at it, no one really thought that cerebral palsy could be genetic. It's actually the most common physical disability in children, so it affects about 1 in 700 in developed countries. And it's an umbrella term for a group of disorders that just affect movement and posture. These disorders result from altered development of the brain or sometimes injury to the brain. The general perception is that it's usually only injury to the brain. CP is usually non-progressive, that's the movement, but it's not unchanging. And so this nice little infographic down the bottom of my slide just shows you some of the things that come along with cerebral palsy. So there's the movement, so unable to walk or unable to talk, but more than 3/4 of people with cerebral palsy also experience chronic pain. About 1/4 of them have epilepsy, and about 1/2 of them experience learning impairments as well. And the reason why we really love beta-catenin is that it's helping us in order to send the message that cerebral palsy should be a trigger for genetic testing. So in many countries where we have quite good genetics healthcare system, if you have intellectual disability, a global developmental delay, or an epilepsy diagnosis, that's a trigger straight away for a genetics referral. And those of you who have been through that diagnostic odyssey know that for many people that genetic result is very helpful. This whole community is together because you all have that beta-catenin in common, which is I would say generally useful. We know so of these individuals, about 50% of them will receive that genetic diagnosis. If you get a diagnosis of cerebral palsy, however, it's typically not a reason for a genetics referral. So then you get sent into a different healthcare pathway, typically rehabilitation or physiotherapy, and then you're moving further and further away from the people with genetics experience. But we know from our research that about 30% of those individuals who get a cerebral palsy diagnosis could actually get a genetic diagnosis as well. So we want to change this inequity. So over the last 7 or 8 years, our group and that of others have been monitoring the genes that are associated with cerebral palsy. So now we've got up to a count of about 190 genes reported, and these are in at least 3 unrelated families and often from different groups. And when we look at the genes which are associated with cerebral palsy, we see that it's not much different to genes which are associated with other neurodevelopmental disorders. So that could be intellectual disability, epilepsy, or autism spectrum disorder. And you can see the numbers of genes in this diagram here overlap with each other. So this means that these neurodevelopmental syndromes are not just one item. It's not just intellectual disability or just epilepsy or just autism. They're a mix of things, and it's much more complex. If you want to know where beta-catenin sits in this group, it's in it can be called a cerebral palsy gene, it's been called an autism gene, and it's also been called an intellectual disability. So our interest really picked up as a result of our studies, but also this very large study involved about 1,500 individuals who had been diagnosed with cerebral palsy, but typically they were referred for genetic testing for another reason, such as intellectual disability. But when they were retrospectively analysed, about 1/3 of them had a variant which would cause that disease, so that could explain and give them a diagnosis. And of those, 4% of those individuals, so out of 450 individuals that had a genetic diagnosis, 4% of them were actually beta-catenin. This actually makes beta-catenin the most recurrently affected gene in people who have a diagnosis of cerebral palsy. But if they're getting referred down the wrong pathway, there's probably additional people who've been diagnosed with cerebral palsy that aren't getting a genetic diagnosis. So our research questions, I'm going to go into the science now, hopefully it'll be accessible, as how frequent are beta-catenin pathogenic variants? And we wanted to know whether cerebral palsy was part of the syndrome or it was something additional. And to do that, we did a retrospective study and asked what has been described today. And I'll go through some of our results from that paper, which was published last year. And we then wanted to know, and Wendy's touched on this also, how do beta-catenin variants actually cause the disease? And we also wanted to see whether we could model the syndrome in a cell model. So these are the things that I'll follow up with now. So first thing we wanted to know is how frequent are variants. So when we started this study, there were actually only 33 people who had been described in the literature. And they'd been recruited from all over the place. So intellectual disability studies, general neurodevelopmental studies, autism, eye disorders, movement disorders. At the same time, the beta-catenin registry came to my attention. And this is a little snapshot from the website from 2019. And all of these dots, all of these people, there were more than 100 people in that registry. So I asked the question, like, how come there's only 33 in papers that we're reading, but there's more than 100 people who have been genetically diagnosed? I actually just quickly snapped that map last week when I was putting this final touches to this talk, and I see there's a lot more people that are signed up now, which is great to see. So there were two ways in which we went about this. The first one was to use this matchmaker exchange network. So this is a network between scientists, doctors, and also patient groups or people living with these disorders who can submit safely their genetic information without submitting any personal information. So you're protected from being identified, but it allows people to become aware that a variant exists in a particular gene. So this is phone a friend for geneticists. Through using this network, we recruited additional patients and made contacts with all of those people that I put on my acknowledgement slide, which I don't think anyone saw. And then we also used traditional methods. So we used scientific databases, one of which was ClinVar. And ClinVar is where a lot of these large genetic testing companies and also private or government labs submit all of their genetic variants. Again, it's consented and anonymous. And then we also did the hard graft, which was just read all of the papers. And these aren't papers that were just associated with beta-catenin, but also papers where people had just sequenced cohorts of people who had neurodevelopmental conditions. There's one final part of this, which is the magic ingredient, which is Dr. Kayumi, who really did a lot of the hard work to pull all of this information together. And what Sayaka found is 404 individuals throughout all of these resources. And they represented 392 variants in beta-catenin. So some of them occur over and over again. And they're all likely causing beta-catenin syndrome or the related syndrome FEVR, which is a familial eye disease and usually doesn't come with intellectual disability. So we included all of these. So the first thing we wanted to say is because all of these, the people we were aware of had been recruited to so many different studies, we wanted to ask the question, is beta-catenin a gene which is associated with lots of different disorders? Or is beta-catenin a gene which is associated with one disorder, which lots of people give different names? And so this data show you that, in fact, the second answer to that question is correct. So all of there were 9 traits which we saw were quite commonly mentioned in all of these reports that we were able to identify, and that was in 120 out of the 404 individuals that we'd identified. And what you can see, the red bars mean that a person has one of those particular 9 traits. And what you can see is most of the people have mostly in the red category rather than in the blue category, which means they don't have that particular trait. So what that means is essentially, if you have one of these symptoms, it's more than likely you've got at least 2 or 3 other of these symptoms. And it means that the disorder looks pretty similar across multiple people, at least what's been described today. What I was pleased to see is when I looked in the beta-catenin survey data, which is from 2020, and there's 104 individuals who have signed up, and there may be some overlap between these 2 data sets, is that actually if we plot out just the red and the blue across these, that we actually see it quite similar. So our results are quite consistent with other independent research as well, which is good to see. So we go back to our question of whether cerebral palsy was part of this disease or whether there was an additional brain injury which was causing some individuals to be diagnosed with CP as opposed to beta-catenin syndrome. So here we have people on the left who have been diagnosed with cerebral palsy and people on the right who did not get a cerebral palsy diagnosis or wasn't specifically mentioned. And what you can see in general, if you look between these two, that the percentages of each of these traits which are being observed are pretty much the same across the board. There's a slight difference in the motor-related or movement-related phenotypes, and that's perhaps not surprising because you absolutely have to have at least one of these traits in order to get a cerebral palsy diagnosis in the first place. Overall, we believe cerebral palsy is part of the diagnosis. It's perhaps not specific enough, but it's also not incorrect to describe the movement phenotype of beta-catenin syndrome as CP. We wanted to know how people were also getting their genetic diagnosis and how long it was taking. So of those individuals, we had 79 where we could track their genetic diagnostic odyssey. So most people receive a standard newborn screening test, and that's a very valuable and proven method of identifying really damaging conditions. Although in the case of beta-catenin syndrome, which is not captured in this kind of screening, it's not usually that effective. So only 3 out of those 79 had a result from that initial test, that first frontline test. What we saw then is that most people got referred for like a specific gene test. Quite frequently that was Angelman syndrome, which looks phenotypically very similar to beta-catenin syndrome, except for there's a lot more epilepsy in Angelman syndrome. When these are usually unsuccessful as well. If you don't pick the right gene, you don't get a result, and less than 5% of individuals got a diagnosis from that. Most people got their diagnosis from either whole exome sequencing, which is sequencing all of the gene content of our genome, or gene panels which capture all of the genes which we know currently to be associated with disease. So what these analysis told us is that we should cut out the gene-specific tests and move straight into exome or genome sequencing when it's uncertain after a frontline test. So now I want to talk a little bit about beta-catenin biology. And I've used this analogy. I'm not sure whether I'll regret this later, but I ended up having a lot of pigeon theme later on in the talk because of this. But beta-catenin is a really cool protein or gene in that it's a multitasker, which is not typical for most genes in the genome. So it first acts as a signaling molecule. So there are small proteins called Wints, which travel between cells, and they carry a small message with them. And beta-catenin is like the receiver of that message. When it gets that message, it moves into the cell nucleus, it goes to where the DNA is located, and it activates the synthesis of some genes. On the other hand, beta-catenin is independently associated with cell-cell adhesion, so links between one cell and another. There are proteins called cadherins which stick out of cells and hold on to each other. Beta-catenin is the anchor on the inside of the cell, and it locks it into the cytoskeleton, which is like a mesh. If you imagine a big ball of chicken wire inside your cell, that would be a reasonable analogy. And that holds the cell together and gives it its shape and strength. So Wendy has shown, I think I've got a different color scheme. So all of those tricky variants, which Wendy was just talking about, are in yellow in here. So one of the things we were worried about, because beta-catenin prior to 10 years ago was predominantly associated with cancer. So we wanted to know straight away whether beta-catenin variants which cause the syndrome have any relationship to the cancer-causing variants, because that could be a risk later on in life. So we know that from many years of research that beta-catenin variants which cause cancer are overactivated. So it's like having way too much Wnt signaling message. Here you can see that lollipop plot with the protein structure along the bottom and the distribution of variants. And you can see that cancer variants cluster very, very frequently at the start of the protein. And this part of the protein is what keeps beta-catenin stable or unstable. So when it's there, you usually don't have much beta-catenin. When this is interfered with, this beginning of the protein, you end up with a lot of beta-catenin. It becomes very stable and you get too much message. Now, the good news for the neurodevelopmental disorder is that the face of those variants is very different compared to the cancer variants. So here you see the cancer variants in this pie chart, everything's yellow. Over here, everything's red, blue, or purple. And all of these types of red, blue, or purple variants are ones which cause loss of beta-catenin protein rather than gain of it. We also looked in the general population, about 16,000 variants which have been identified. What's curious about those is that they change at the DNA level, but they don't change the protein sequence. So all of these variants mean that beta-catenin is usually highly protected from having any changes at all. There were still in this collection some variants which have a similar colour to the variants which are found in cancer, and these were the ones we were interested in. So we tried to ask the question, can we work out more clearly what these variants are doing, because they are the ones which are usually uncertain, and those are the ones which cause a genetic diagnosis to be not to be made. So here we had a way of measuring Wnt signalling, and here we use an assay where we created a gene which has a promoter, which has a section of DNA which attracts beta-catenin, and when beta-catenin is close by this gene, it turns on a gene which creates visible light. It's actually the same gene which is found in the back end of a firefly. When that beta-catenin is damaged or doesn't work, that light is turned off. Okay, so here's some results from that. In grey, we have our the protein as it's usually found in most people, that unchanged protein. And when we add that unchanged protein to our assay, we turn the light on. So this protein works. When we look at variants in the gene, which are just found in the general population, but change the protein sequence slightly, they're also on. When we looked at a variant which is associated with the non-syndromic eye disease, FEVR, that also has pretty much the same activity as the normal protein. When we look at 3 variants which were associated with beta-catenin syndrome, 2 of them completely wipe out the activity of this assay. Curiously, 1 of them didn't. When we mixed these proteins together, as Wendy said, usually beta-catenin has 1 good copy and 1 which is asleep, we find we get almost the same result. So all of those proteins which normally don't cause the syndrome have normal activity. Those which cause the syndrome, 2 out of 3 reduce the activity, 1 did not. And that's very interesting. That's telling us that Wnt signalling is sometimes affected, but not always affected. So the last thing I want to go through, I'm not sure how fast I've been going or slow, is the model we're trying to make of the syndrome. So Wendy also mentioned stem cells. We elected to make a model that way, and we chose to make our own rather than get a patient donated stem cell line. So stem cells are very clever in that they can self-renew. So you can make another copy of a stem cell from a stem cell, but you can also turn a stem cell into lots of different tissues, including a neuron or a brain. So here we use technology which is able to cut the DNA, and here we cut a big chunk out of the beta-catenin sequence and then stitched it back together. And once that chunk was removed, we actually lose the beta-catenin protein. So with our cell model, we end up with a type of cell which has all of its beta-catenin intact. We call those wild type. We have those which are the model of the disease, which is half of the beta-catenin. We call those heterozygotes or hets. And then we had a cell line which had absolutely no beta-catenin at all. We call those knockouts. So just down here is a little bit of biological data which shows which is measuring the beta-catenin protein in these cells. Every time you see a little black smudge on this background, that's measuring the protein. See, in the wild types, we've got a certain amount. In the hets, we had about half, and in the knockouts, it's completely gone. And down here is just another protein which is unrelated. When we put these cells together in cell culture, you leave them just floating around. They tend to want to stick to each other, and that's part of that cadherin network which I mentioned. So again, the wild type and the heterozygotes form these big clumps, but our knockout cell lines, which don't have any beta-catenin at all, fell apart. They lose that connection. The last thing we've done, this is very early work, is to look at how these cells turn into neurons. What we've found is these knockout cell lines turn into neurons much faster than the other 2 types of cells. So in this image, you're seeing red cells, and they're the cells which you create prior to forming a neuron. You also see these wispy, long green cells. Apologies to those who may not be able to distinguish green from red. This is natural colours that come from the microscope. I can't change them. You see the cells have different shape as well. So these big bulky cells are these neuronal precursor cells. And in order to make a brain, you need a mix of those cells before a neuron and neurons themselves. In this situation where you push every cell into a neuron, you run out of cells to replenish and create and maintain your brain structure. So this is frequently seen in other neurodevelopmental disorders where microcephaly is a feature, so smaller head size, and that's a major feature also of beta-catenin syndrome. So this is quite encouraging that our model follows along the biology as we see it. We also see it at a biological level that even though under the microscope these heterozygous cells look like wild type ones, at the level of their RNA, they look partially like the knockouts. So we do have a good model of the heterozygous disease as well. So just to conclude, we have a loss of function of beta-catenin, and they cause a relatively similar syndrome in most people, and that includes impaired movement, which can be classed as cerebral palsy. We have now a method which can test the functional effect of at least some MNCS changes, and these can be important for providing a genetic diagnosis. And we also have a human cell model of beta-catenin syndrome, and we see changes during early neuron development. I would note any researchers that are online today, if you wish to use this resource, please get in touch. We're happy to share. This gives us potential to understand the biology and also development and testing of potential treatments. So thank you very much for your time, and hopefully that was what you were seeking.
One thing led to another. Here I am speaking with you and being a member of the scientific advisory board, and hopefully working towards developing a new treatment. I should just mention I am affiliated with Boston Children's Hospital. I've been there for over 30 years, the last 20 years on a part-time basis. I decided to go to industry relatively early in my career to work on new therapies for rare genetic diseases. And I started my career at Genzyme in 2000 and had an opportunity there to work on 6 drugs for rare diseases, all lysosomal storage disorders that were approved. And these are treatments that are given either intravenously every 1 to 2 weeks or small molecules given once or twice a day. And in the mid 2015 time period, I became really enamored by some of the advances taking place in the gene therapy field, especially with genome editing and CRISPR. So I went to Editas Medicine, which is a Cambridge company that was interested in treating a host of rare diseases as well as cancers using gene editing. And I was involved with the investigational new drug application that was associated with the first in vivo delivery of a CRISPR-based treatment. In 2018, I really wanted to focus on rare diseases and do more than just genome editing. A lot of the gene therapies are available in the US and Europe, but not so much in other parts of the world. And so I went from chronic therapies to one-time therapies, now thinking about market access. And as part of that, I spent about half my time now as a consultant working with a company called Cambridge Pharmaceuticals based in China, where rare diseases literally did not exist before 2018 because there was no definition of a rare disease, and they were pretty much an ignored population. But Cambridge is interested in both developing as well as importing innovative biotech treatments to China. And as part of that, I've been involved in a couple of approvals for rare diseases in China. And I've also, in addition to my consulting work, I've been involved with some different patient advocacy organizations. NTSAD is the oldest patient advocacy group. It's like 65 years old this year. I'm on their national board of directors, and now CTNNB1. So kind of come full circle from being a clinician to a researcher to a drug developer, now patient advocacy and market access. So back, even when I started in rare diseases, each rare disease was really its own entity. There was no commonality that people found. And so because each disease was so rare, they were really almost all invisible. And in 1983, Congress passed what's known as the Orphan Drug Act. So if there was a disease that affected fewer than 200,000 people in the US, then these drugs could be qualified as orphan drugs. And with that came different incentives for companies to develop treatments, like having market exclusivity for 7 years, where no generics could enter the market. There were tax credits, waiver fees, and the like. So that really spawned a revolution in treatment of rare diseases. Before 1983, I think there were like 40 drugs approved for rare diseases. And since the introduction of that act, now there's over 500 treatments. Well, 500 treatments sound like a lot, but we know there's probably about 10,000 genetic diseases that have been defined now, especially through whole exome sequencing. And with all those diseases, it's been estimated there's about 30 million people that are affected in the US. So if you take that as a percentage of the population, it's almost 10%. So you can see that's actually a big lobbying base. And there are some now national organizations like NORD, National Organization of Rare Diseases, that act as an umbrella for all rare disease groups. And with that comes a lot of power and strong voice. So people say, individually rare, but collectively strong. And I think that's the case. So my talk, I'm going to focus mostly on drug development for rare diseases, because it is a bit different than developing drugs for common diseases. But this is kind of the general timeline. There's two main halves, like before going into patients and then after going into patients. Before going into patients, that's the discovery phase. That can take 5 to 10 years. You have to define what it is that you're trying to treat at a molecular level. How are you going to approach it? Is it a small molecule? Is it gene therapy? Once you develop a treatment that you think is having an effect, now you want to fine tune it. Maybe if it's a small molecule, you want to make it last longer in the body or distribute to the brain more efficiently. Then you need to test it in animals to make sure that there's no serious side effects and that the drug is actually doing what you think it's going to do. And then along with that, you need a way to manufacture the drug that's very consistent and safe as well. Most of drug development does involve small molecules, which is basically a series of chemical reactions to make the drug, but some of the more advanced molecules, the nucleic acid therapies, the gene therapies, those are much more complicated, and that can take quite a while to figure out the process. But once you get to the point where you feel like you have a drug that's ready to go into patients, you've done all the animal work, and you have a clinical trial protocol that will be used to investigate the first group of patients, then you can go to the FDA. The FDA is the gatekeeper of all drugs. So they oversee drugs that are approved, and they oversee drugs that are still in clinical trials, or what we call investigational drugs. So we have to apply to FDA. They actually have a pretty quick review period of 30 days. After 30 days, they can give us a green light to go ahead in patients. And then traditionally, there's different phases that drugs go through, 1, 2, and 3, where we start off with small numbers of patients and then gradually work our way up. In each phase, we're asking a different question that ultimately will take us to the point where we can show that our drug is safe and effective in a given patient population. And drugs are approved for specific indications or specific diseases. Sometimes there's drugs that can be used for many different diseases, but the FDA will want you to test them in all those different diseases if you want to market it for that. And then once you complete the trials, which on average can take 5 to 7 years, there are ways to accelerate that by a couple of years. Then we submit a package to the FDA. Last package I submitted to FDA had 330,000 pages of information. So they can't do a 1-month turnaround on that. But if you're dealing with a serious disease, they move up their timeline to about 6 months. And so normally it's about 12. And then at the end of that, they tell you whether or not your drug is approved and can be marketed. Now, when you think of drug development, in the end, it does cost a lot of money because there's a lot of failure along the way. In fact, drugs that go through all that research testing and find their way into the first human clinical trial, then we have on average about a 10 to 15% of actually getting approved down the road. And you can imagine as you go through development and start getting better and better data, then that probability of success goes up. So the 3 different studies, 1, 2, and 3, because there's so few patients who have rare diseases, we tend not to do 3 independent studies if we can avoid it. And we usually combine the first 2 or the last 2 studies. And the first study is really to investigate, what is the drug doing in the body? Is it causing any side effects? How high a dose can we go? What dose of drug do we need to achieve the intended levels? So in the case of CTNNB1, if you're looking at beta-catenin levels and you have a drug you think increases it, you want to give progressively higher doses until you can monitor that the level is back up to where it's supposed to be. We also look at what we call the pharmacokinetics. How long does the drug last in the body? If it only lasts for 12 hours, then we know that's a drug we probably don't want to give once a day or once every 2 days. We want to give it probably twice a day. You don't need to give it 4 times a day. So that's the kind of information we need to know. And then how is it excreted from the body? Does it get metabolized in the body, converted to an inactive substance? Does it go out through the urine? Does it go out through the stool? People that have kidney disease, may develop high drug levels if your drug is eliminated in the urine. So all of that we need to figure out in that first study. For small molecules, we usually start with healthy volunteers. It's usually college students who want to make some extra money for participating in the study. We usually use a relatively small number of people, 10 to 30 individuals. But for some of the more advanced therapies, like protein therapies or gene therapies, we'll go directly to the patients because you wouldn't want to give a healthy volunteer a biologic that might induce an immune response or hang around forever in the case of a gene therapy. So then if we combine those studies, in addition to characterizing what the drug is doing and how it's being metabolized by the body, we want to know, is there any sort of efficacy that we can monitor in patients? Are there any changes occurring? A lot of times we look at biomarkers. These are either lab values or imaging endpoints that can tell us objectively if the drug is doing what it's supposed to do. But we'd also like to get an idea of, are we seeing any clinical changes in the patients? And that, usually requires somewhere in the order, 20 or 40 patients, because every patient may behave a little bit differently. You may see a bigger effect in one patient, not another. It could be that whatever you're looking at, let's say it's speech delay, one patient may have speech delay that's very severe, you don't see much of an effect. Someone else may have moderate speech delay, you do see an effect, and someone else may have normal speech, and you wouldn't expect to see any change. So you need to really evaluate how your drug is impacting the patient, and ideally identify some endpoint, some measure that the drug is providing some clinical benefit, and carefully select your patient population. And that's when you get to your phase 3, or what we often call a pivotal trial. This is the one that the FDA is going to look at and judge if your drug is safe and effective. And with that, we need to have enough patients in the trial that we can pretty confidently say that the drug is working. And that's where statistics comes in. You may have heard about a p-value less than 0.05. That's the probability that your effect that you're seeing is occurring by chance less than 5% of the time, which means that it's really related to the drug 95% of the time. And that's the criteria that FDA likes to use. They don't like to do studies or evaluate studies that don't have a comparator, because some of these endpoints are effort-based, and just being in a clinical trial and getting good care, sometimes patients will perform better even though they're not getting a treatment. And so having another group as a control, either taking placebo or maybe taking standard of care active drug, you'd like to be able to compare how your drug is performing based on the current standard of care. And it's also an opportunity to evaluate safety. Again, if you know you're getting a drug, you may imagine that you're having headaches or nausea, but being able to give a placebo at the same time tells us whether those side effects are really related to the drug or really just maybe some nervousness or anxiety as being part of a clinical trial. And these studies, generally are 40 to 100 patients. Wherever possible, FDA likes to collect at least a year of data on 100 patients. We know that in cases like CTNNB1 is probably not going to be possible because that's such a big percentage of the population. There have been drugs approved with as few as 10 patients. So it really depends on the population. I'd say FDA has become much more flexible in the last 10 years. They really, if you remember Wizard of Oz, where the three main characters go to Oz and they're speaking to the wizard behind curtain, and you have no idea what's behind the curtain. That's the way it used to be when I started off in drug development, interacting with FDA. We never knew what they were thinking. And then, let's say in the early 2000s, it really became both a patient advocacy movement as well as industry movement to have more transparency from FDA. And so suddenly those curtains became open. We began participating in the same conferences. FDA would explain why they felt certain ways about certain types of trials and data. And it was much more of a shared understanding of what it would take to get a drug approved. And I think from their end, they've also evolved. They now have these things called listening sessions where patient organizations can apply to FDA and have both physician explain the disease as well as some patients and their families explain what it's like to live with the disease, so that when FDA does see a drug come through for that disease, they have a much better appreciation of how serious it is and how it impacts people's lives. And that's something that this organization could schedule as well. So what does it mean when you participate in a clinical trial? I think it's important to realize that clinical trials, these are not treatments per se, because they're not approved drugs. We don't know if the drug is going to help you or hurt you. And so you shouldn't necessarily go into a clinical trial thinking that, this is going to be the panacea for my child, because there could be side effects that occur that maybe make you wish maybe you hadn't participated in the trial. So it's important to have an open mind coming in. There's a lot of regulation around clinical trials. We have to go through FDA for approval. The site, the institution that you're going to be going to has its own internal committee, institutional review committee, that reviews the protocol and makes sure that the investigators of the sponsor are not doing anything untoward. They're not removing a gallon of blood from an infant. They're not putting patients through painful procedures. They're not, having so many clinic visits that it's going to be infeasible to conduct a trial. And all of this is explained as you first decide whether or not to join a trial. There's something called the informed consent that explains what the trial is about and what the potential risks are, what the potential benefits could be, and what the expectations are. Because when you're in a trial, you don't just get a treatment and go home. You have to go back to the clinical site and get evaluated repeatedly. It could be every week, for the first few weeks, just to make sure there's no safety issues occurring. Generally over time, it gets spread out to maybe like every 3 months or every 6 months. And then, during the screening phase of the trial, not everyone would necessarily be appropriate for a trial. So again, if you're looking at changes in speech, if someone has normal speech, then they wouldn't be a good candidate for this trial. So there's different inclusion and exclusion criteria to try to select a population that's going to really test the hypothesis whether or not the drug works. Now, for a lot of common diseases, if patients have to travel, it's usually not that far. For rare diseases, there might just be one individual in a particular state, and a clinical study might be taking place across the country. And so the companies realize that this is a big imposition, and especially a financial one. So I would say 99 out of 100 times, they cover all the costs, because they don't want families not participating because of a financial burden. So they'll pay for travel, they'll pay for hotel, they'll pay food while you're there, and usually with a companion, because we recognize a lot of these diseases affect either young children or they can affect individuals in a severe way that need some assistance while they're there. So patients will continue through the trial. For rare diseases, if it looks like the drug may be having a positive effect and there is no other treatment, a lot of times families will continue what we call a long-term extension trial until the drug is approved. And it helps the patients. It also helps the sponsor to collect long-term data, because if you only have 20 patients and you have a 6-month trial, that's not a lot of safety data. But if you have 5 years worth of safety data on a small number of patients, it's much better than just 6 months. So with FDA, especially with both rare diseases and serious diseases where there's no satisfactory treatments, there are a number of pathways that drugs can be eligible for to help expedite the review process. I've listed a number here. Orphan drug designation is something you can apply for as soon as you have a drug that looks like it's having an effect on the animal model. But there's other therapies based on whether or not there's preclinical data or actual clinical data suggesting a benefit that you can apply to FDA. And if they grant that, then it increases the interactions with FDA. They'll actually call you on the phone, which is unheard of without these designation. And one I want to point out here is one called the Rare Pediatric Disease Designation, where this was an effort to recognize that children are a vulnerable population, and there should be some incentives to companies developing treatments for children. And what the FDA had decided to do was to grant what's called a priority review voucher at the time that the drug is approved if there are a number of children in the clinical trial. Well, it turns out that, and I don't think FDA anticipated this, those clinical trial vouchers, they reduce the review time from about 12 months to 6 months. And they can be used for any drug. So a common drug that would normally take a year, if you have a priority review voucher, FDA has to review it in 6 months. So if you're looking at these big blockbuster drugs, for big pharma companies, they're very interested in getting things accelerated. And so these priority review vouchers can actually be sold. I mean, they can be given away for free, but they actually have a lot of value. And there's probably been about 30 that have been sold to other companies. And the going price is about $100 million. So that could actually fund your entire development program. So I think for ultra rare diseases where you may not think that there'd be enough profit to keep a company sustained, there is a potential benefit of getting one of these vouchers that is worth $100 million. So when the FDA reviews products, they balance risk and benefit and overall safety. So if you're dealing with a skin condition that's not that severe, and there's some serious side effects, the FDA may not approve the drug. But if you have those same side effects for a fatal disease, FDA will say, well, we know what the alternative is, so we're willing to accept that risk. And I'd say they've become more open to patient and family input, especially with, for example, Duchenne muscular dystrophy, where there's very strong patient organizations in that disease, and they lobby very heavily at FDA to get that on the list, and that we are willing to accept a lot of risk to save our children, because we know what will happen without treatment. And the FDA, they feel like it's their responsibility to protect the public. They do a very noble service, but when they heard the families who were willing to take that risk, they thought to themselves, well, if they're willing to take the risk, why aren't we? And so we're seeing a lot more approvals, I'd say, for rare diseases, even ones that could have some serious side effects. So why are rare diseases so popular nowadays? Well, it began with the Orphan Drug Act, but as we learned about genetic diseases, we could characterize how the disease, once you start with the gene, we could characterize how that disease actually manifests in the body. And to researchers, it could suggest different ways of treating the disease. So this is an example of a lysosomal storage disorder where there's a deficiency of an enzyme in an organelle called lysosome, which is like the cell stomach. When the enzyme doesn't work, that substance builds up and it causes the cells to become sick and not work properly. Well, if you know what the underlying gene is, there are ways to correct the gene, either gene therapy or gene editing. If the protein isn't folding properly, people have thought of developing small molecules called chaperones to help them refold. The company I worked at, Genzyme, we just went ahead and whatever enzyme was missing, we just gave it back to the patients. There's other methods you can think of because all these enzyme pathways have a forward direction and a reverse direction. So if you have something accumulating in the body, instead of trying to break it down faster, you can actually produce it more slowly. And so one of the drugs I worked on had that mechanism of action. And so you can see that there's many different opportunities to intervene with several of these rare diseases if you understand them very well. Because of that, people have found that when you compare these orphan diseases, where usually the disease understanding is very good, more common diseases where there might be lots of different environmental as well as genetic factors, that the success rate going from the initial drug application to approval is about half. And again, 50% higher approval rate than common diseases. The other reason why companies like rare diseases, because usually we do have fewer trials that are smaller. So the length of development is about 1.5 years shorter. And then because many of these are serious diseases, the FDA has these expedited pathways to review them faster. So we're looking at faster and cheaper and better understanding. Now, I suppose it goes out saying that these drugs are not cheap, because the way that companies figure out what to charge is they look at the size of the patient population, how much money they need to make back to recoup their investment and have a profit. And if you're dealing with a large population, the drug doesn't have to be that expensive, but for small disease populations, that ratio becomes much bigger. And so on the x-axis here is a disease prevalence, which is how many patients actually have that disease. And you can see that for the more common rare diseases, those that are above, say, like 10,000 people, the annual cost could be anywhere from like $25,000 to $100,000. And there's a lot of cancer drugs that are in that price range. But as soon as you get below 10,000, you see the price goes up and exponentially. We have some drugs that are $900,000 a year. So that's pretty crazy high. And I'd say, some of those are chronic therapies, some are gene therapies. I think the most recently approved gene therapy is now like $3.5 million, but it's a one-time treatment. So that's something we're going to have to contend with, because in the US, US market will support these prices, but Europe is pushed back, and certainly outside of US and Europe, other countries aren't going to pay these kind of prices. So I said the good things about rare disease. What are some of the challenges? Well, most obvious one is that there's very few patients that can be available to participate in clinical trials. So setting up registries, setting up patient organizations that can bring people together, doing high-risk screening. We heard about the CP clinics. There are ways to try to enhance the number of patients that could be used for trials. Natural history not being well known. If you go to FDA and say, we want to look at a change in XYZ, FDA is going to say, well, why do you think that's important? So you need to know that whatever it is you're measuring in your clinical trial is important to patients and their families, but also it's well characterized. So all the effort that's going on here, to collect natural history data is really important. Many of these diseases, because they haven't been studied very well, people don't know what am I supposed to look at in a clinical trial to show a drug is working. So that's where this natural history study can actually produce some new endpoints that could be very useful for trials. Pediatric considerations, if you have infants, they're going to have different endpoints than, say, an adult. You're going to have limitations in terms of what you can do, cooperation, amount of blood that you can take, slow disease progression. A lot of the neurodegenerative diseases, they decline over periods of years, but clinical trials take place over months. So how are you going to show a difference from the natural history of the disease? So you either need a disease that is very aggressive, that declines very rapidly, or your drug has to show some sort of improvement in the people getting it. You need to see a difference between those groups. And then finally, diseases with serious and high unmet medical needs. Some of these diseases, patients may not survive through the clinical trial. There may be patients that want to get into a trial, but they're too severe, and yet there's no alternative. So companies have to deal with that as well. And sometimes after enrolling a clinical trial, they may offer another program called expanded access program. That's not really looking to show that the drug is effective, but really more like a compassionate use to help people that aren't eligible through the trials. There are some things we can do in trials to make them more efficient to accommodate small patients. Certainly, if we have a choice of a number of different efficacy endpoints that we're going to look at, taking the one that has the biggest effect makes a lot of sense, because the bigger the effect, the fewer the patients you need. You want to be able to match a particular endpoint with either the disease stage or the severity of the disease. Not every trial has a placebo control. I'll show you some examples. Enriching patients. So we're again looking at speech delay. You want to make sure everyone in the study has speech delay, otherwise you may not see an effect. And there's ways of combining trials, and then the accelerated approval pathway. Maybe your disease is so slowly progressing, there's no clinical events you can record, but you know that change in the level of something in the blood correlates very well with long-term clinical benefit. So there are ways through the accelerated approval pathway that drugs can be approved on the condition that down the road show that there's some eventual benefit. This is an example that compares effect size with number of patients needed to show a statistically significant effect. And, for people that have a little bit of a math and stats background, you're basically looking at the number of standard deviations of change on the x-axis. And anything above 0.8 is considered a very large change. And on the y-axis, the number of patients per group that you need to show a statistically significant result. So if you had an effect size of 1, you need about 16 patients in your drug-treated and your control group. And that is doable for rare disease, for most rare diseases, but an effect size of 1 is really big. So you need to make sure through some of your natural history data that, and some early clinical trial data, that you think you're going to have that big of an effect. Now, FDA has become increasingly more accepting of non-placebo studies using external controls based on natural history data. So all the data you're collecting as part of the conference is very important. Sometimes this can be data collected in the past, or it can be data collected while the trial is going on. It could be different individuals. There could be data collected before treatment in a child, and that to data collected after treatment in a child. What the FDA raises concerns about with these types of designs is that there's no randomization. So there's no patients who are only getting a drug versus those who are getting placebo. Everyone knows if they're getting the drug. And again, if you're looking at something that's effort dependent or how someone feels, there can be these what we call placebo effects that occur in trials where it's not really the drug itself that's responsible for the effect. So unless there's a huge difference, the FDA really encourages people not to do these kind of studies. There could be a drug that is approved for your particular disease, but it doesn't work, say, all that well. But you could compare your new drug to the old drug, so everyone is getting some sort of treatment. And FDA does like those kind of studies, because now you're randomizing patients between the two. What they typically like are randomized placebo-controlled studies where people coming into the study get assigned to either a placebo or drug, and you don't know, neither the patient or the physician knows who's getting the drug and who's getting the placebo. If your drug has a very short acting effect and there's some kind of immediate change you can monitor, you can use these crossover studies where everyone is taking drug for a period of time, you see what happens, then they switch to placebo, and you see what happens or doesn't happen, and you can compare those changes within the same trial. And then the N-of-1 studies, which you heard of earlier today from Wendy, these are really interesting designs where, again, if you have a short-term effect of a drug, you could assign different treatment sequences, either drug or placebo, over certain periods of time within the same patient. Look at what happens while they're on drug versus placebo. And I put this up as an example that FDA uses, that you don't need many patients in this type of design. In fact, they approved a drug called Danazol for a disease called hereditary angioedema, which is a genetic condition that leads to this periodic swelling, almost like hives that can occur in the skin, can occur in the throat, very painful. And these are events that last for like several hours to days. And what was done back in the 70s was this N-of-1 study, which is 9 patients. And so each patient, unbeknownst to them, receives this randomized treatment sequence over a course of a year, where every month they would be either on drug or placebo, and it would be some random order. And while they're receiving treatment, if they had one of these attacks, then they would move on to the next treatment in the sequence. And then they would go for up to a month. And if nothing happened, then they'd move on to the next treatment sequence. And what the investigators did was they reported how many of these events happened while patients were on placebo versus receiving drug. And what they found, these 9 patients, was that 94% of the time when the attacks occurred, patients were on placebo versus just 2% for drug. So it was pretty dramatically different. And what FDA would say is, it doesn't really take statistics to see that big difference in self-evident. So again, 9 patients, they found a very creative way to show a drug has an amazing effect without a lot of patients. And so the last, I think, thing I wanted to touch on was that if you do have these diseases that are very slowly progressive or non-progressive, and it's difficult to show a clinical benefit, FDA does have a separate approval program called the Accelerated Approval Program, where if there's a laboratory change or imaging or some physiologic measure, maybe it's an EEG, that correlates very well with disease status, then the FDA may allow that to be used to support an approval, as long as the company agrees that down the road they're going to conduct a study. Maybe it's going to take 5 years if you're looking for clinical benefit, or maybe you have to go in a very young patient population to see clinical benefit. As long as the company agrees to that, then you can get a conditional approval, get it reimbursed by insurance companies. And there have been several examples of that occurring, including this past week, last Friday. There was a new gene therapy approved for Duchenne muscular dystrophy. That's an accelerated approval. What that does is it speeds up the timeline by several years. So again, we have a new FDA. So finally, just to summarize, even though these rare diseases have challenges, usually our understanding is much better than for common diseases. So our probability of success getting a drug through development is higher than for common diseases. Placebo-controlled studies, I know families don't like them, but if done, within a reasonable time period, that's really the gold standard that FDA looks for. And again, I think with treatments that are reversible, there could be situations where patients receive both placebo and drug for different periods of time in the same clinical trial. That would be very powerful that FDA would accept. And for for rare diseases, we do have these expedited regulatory pathways that can off, 2 to 3 years off development. So I think, there's a lot of hope for rare diseases, definitely on the radar down at FDA. And I think everything the organization is doing, in terms of being clinical trial ready, is spot on. And as soon as the drug becomes available, I think taking the data that's been learned in natural history and trying to come up with a creative clinical trial design that won't be too much of a burden on families, won't be too long, and will show very definitive results. That would be the goal. I have to close there. Thanks.
Because we're running a little bit tight on time, we're just gonna skip that Q&A. If you have specific questions, you can email me or the info@CTNNB1.org, and we'll direct those questions accordingly. The next speaker that is going to be with us today is Mary Cobb, who is with Global Genes and Rare X, and we're very excited that she's with us today. So our next presenter is Dr. Scoles out of the Children's Hospital of Philadelphia, who's going to be talking about preventative eye care for our children. And then after that, we'll have the Tufts team speaking. So just want to introduce you and thank you so much for coming.
Thank you. Thank you, everyone, for having me. Lauren and Sean, amazing. This is really a very powerful thing for me to learn from all of you, and also to put all this information together so we can forward our understanding of CTNNB1. So it's a privilege to speak, but it's also a much bigger privilege to take care of some of your kids who I've met and some that I'll meet in the near future. And so I'm going to put up a slide that we've already seen this morning, just to highlight one particular thing, which is that eye abnormalities are very common in CTNNB1, and the severity is on a wide range. We don't really know, based on the genotype, phenotype, relationship, like we've heard this morning, who is going to get the severe eye disease and who's not. That's something that's yet to be sort of borne out. And whether or not kids who are born with a little bit of an eye problem are going to get much worse, we don't know that either yet. We will, and we'll learn a lot more from meetings like this in particular. But I just wanted to share, this is a slide from the group in Slovenia. So if you break that down, this is from their paper also, the most common eye abnormality in kids with CTNNB1 is strabismus. And so strabismus is the group that includes esotropia and exotropia. So esotropia is the eyes cross in, either one or both. And exotropia is one or both of the eyes go out. And that's very common in CTNNB1. This is a paper, the Kayumi paper that was cited by the Australian group. And you can see in these pictures here, the vast majority of these kids have some strabismus. And it's something that can be addressed with surgery. But it's important to realize that strabismus is not a cosmetic problem. It's a visual development problem. If one of the eyes is in or out, the child won't see with that eye, and you can't get that vision back. So it has to be addressed immediately, not just so that the kid has straight eyes to look better, but so they can see and develop better. So that's, I don't want to de-emphasize strabismus later on in my talk when I talk more about fever. Needing glasses is also common in kids that have CTNNB1. So these three categories together, hyperopia, astigmatism, myopia, that encompasses a bunch of kids with CTNNB1. You see it's common. And if you look at the kids that are here, a lot of them have glasses. And so that's something that all of you have lived and have seen. And again, it's not just they're a little bit blurry, they need glasses to see a little bit better. If they don't have a sharp image, they won't develop vision. So you need to get on top of this at an early age so the kids can see for the rest of their life. It's not just that, glasses or not. And then what I'm most interested in as a retina specialist, and that's my bias, so you should read into that as I continue to talk, is fever, because that's something that I see unrelated to CTNNB1 a lot, and then related to CTNNB1 more and more. So I want to spend a little bit of time just going through the basics of fever, so it's not a sort of a black box for everyone, because we hear it a lot, but we don't talk about sort of the basic details that often. So this is a picture of the back of an eye, an adult who's in clinic getting a picture in one of our cameras. And I just want to highlight a few things. There are some blood vessels in the back of the eye within the retina. The retina itself is nice and smooth. It coats the back of the eye like film. And that's what helps you develop a nice clear picture, to see the world. There's also the optic nerve here. That's just a landmark. And I'm showing you that so that when I show you pictures of fever, you'll sort of see the difference. And then this is the picture that's from a fluorescein angiogram. And you can immediately see that the blood vessels, the artery and veins, really pop out in a fluorescein angiogram. And that's why we do the image, is because it gives us better contrast of the arteries and veins. And so abnormalities stand out quite a lot. In most kids, you can't get quality images like this in the clinic. Some you can, some you can't. And doing an angiogram is a little bit harder even than just getting a picture in a kid in the clinic. And we'll talk more about that as time goes on. Yeah, we'll worry. There we go. We're good now. You're good now? Yep. Trying to advance. There we go. Okay, great. So anyway, let's just skip ahead a little bit. I want to leave some time for Q&A, so I'm going to go through this a little bit faster. But I just showed a picture of normal arteries and veins. Now I'm going to show fever. And you'll see fever has a bunch of different stages. We don't need to go through the details too much, but what I want to highlight is that the example images for stage 1 and stage 2 fever are showing angiograms. They're not showing color pictures, because to pick up stage 1 and 2 fever, you really need an angiogram, because the findings can be really subtle. When you're talking about stage 3, stage 4, and stage 5, you're talking about various stages of retinal detachment. And those you can see without an angiogram. But the question that remains to be unanswered in CTNNB1, at least, we kind of know the answer in fever, is that if you catch a kid at stage 1 or stage 2 and treat them, can you prevent them from progressing to stage 3 and 4 and 5? And the answer is probably yes, but the challenge in CTNNB1 is that some kids are born with stage 4 and stage 5 disease, and there's nothing you can do to prevent that because they were already born with it. And so then it's a surgical problem, and that's something that I don't have time to talk too much about. But I just want to go very quickly through this slide to highlight one thing, which is that there's a potential that we should expect fever in all kids with CTNNB1. And the reason is that it's very important in the signaling process that's related to cell the Wnt pathway, as we already heard earlier. And you can see that the same proteins that are very involved in fever, have been borne out to be involved in fever for the past 30 years, are all in the same pathway. So there's reason to expect that beta-catenin, since it's in the same signaling pathway, would cause fever, and it could cause fever in all kids in various degrees. It doesn't seem to, but it could theoretically, and that's why our radar needs to be very much on for looking for fever in CTNNB1. This is another sciencey slide that I have here that just highlights sort of why fever happens. If you have cell signaling that's supposed to help you develop the vasculature of the eye in the womb, and then develop the adult vasculature, so the embryonic vasculature is supposed to go away and the adult is supposed to come. If anything in that process gets disrupted because the signaling is off, then you're going to have vessels that shouldn't be there, or you're going to be missing vessels that should be there. And that drives the pathophysiology in fever. It's generally a vascular disorder that leads to other problems along the line. And so that gives us an opportunity to intervene if we can kind of stop this abnormal signaling process that's going to progress to retinal detachment with time. And so I want to talk a little bit more about fever and severity in CTNNB1. I kind of alluded to this. If you look at the literature and what was sort of summarized in the big paper from the Slovenian group, is that the severe fever is overrepresented in published cases because it's so easy to detect, meaning kids who are born with a retinal detachment usually are detected almost at birth. But we don't know a lot about the kids who are asymptomatic. So if you look at all these papers that have been published, there's a relatively small number of eyes, but they're all very high stages because these are the stages with detachments that are easy to catch. And I think that this is not representative of what the fever is by and large in CTNNB1. My cohort is a little bit different and is biased in the other direction. I have a couple of kids who don't have any fever, and then several kids who have milder stage fever. And this is important because I think that catching them at this stage, you could intervene and prevent maybe progression to a later stage disease down the line when they're older, 10, 20, 30, 40. And so I think this is because we are kind of underdiagnosing fever because it's hard to diagnose. So I have an anecdote, and I don't practice medicine based on anecdotes, but I saw a patient who was newly diagnosed with CTNNB1 who had been followed up for a long time by a great ophthalmologist and treated. And it was only under exam, under anesthesia with an angiogram that we were able to actually detect that there was a stage 3 fever detachment that needed to be treated. It was vision threatening for sure, and something that would not have been picked up in the office. And that's because all kids are hard to examine. And kids with CTNNB1, and by no fault of their own, are harder than the average kid to examine in the office. It's just a fact. In fever outside of CTNNB1, it's universally agreed that treating early, identifying early improves outcomes, and so the same should be true for CTNNB1. And an important point is that a lot of times I see kids with one eye who have developmental delay. They have one eye because they lost it to a detachment and they were sort of ascribed self-trauma, meaning that the thought was that they had been hitting their eye. But there are kids out there probably with CTNNB1 with developmental delay who have not been diagnosed, who didn't hit their own eye. They lost it because of a fever detachment. And if we can prevent any of those, that's really a win for all the kids involved. So I'm just rushing through this so I can answer any questions that you have, but I propose a team approach to eye care in CTNNB1. So I'm a pediatric retina specialist, but I work with pediatric ophthalmologists, and I very much value their input. So I sort of silo the care. We need a pediatric ophthalmologist to manage the strabismus, the glasses, the refractive error, if they have any amblyopia and they need patching, if they have cataracts or glaucoma, that's sort of their bucket. And then I come in to diagnose and treat the fever. So I personally think, as much as I hate to subject any kids to anesthesia, that all of these kids probably need an exam under anesthesia and they need a fluorescein angiogram. And whether they need it once in their life or multiple times is sort of determined based on what their exam looks like and what their risk factors are for getting worse. I think that we won't catch fever in CTNNB1 unless we really look for it in this way in most kids. And so what's the treatment? I alluded to this already, so it's just sort of rehashing here, but the pediatric ophthalmologists that I work with, they handle the glasses, the strabismus surgery, realigning the eyes. They handle the patching and anything else along those lines, and then I handle the fever manifestations. And sometimes it just is observation, meaning they have stage 1 or very early fever, and I said, let's just watch this for a year, let's repeat the angiogram. Sometimes laser is needed, and that halts that abnormal signaling that I referred to for the blood vessels to grow in abnormal ways. And sometimes they have a detachment and they need surgery, including a buccal or a vitrectomy. And some of the kids who are diagnosed at birth need these things right away, and kids later on, it's hard to know when they're going to need it. So it's kind of hard to put a typical algorithm on the care for every kid with CTNNB1. So I just want to kind of wrap up here and ask for any questions, but one question that I get a lot, and I'll sort of preface here, is what is the vision like in CTNNB1? What can we expect? And I'd say that generally speaking, if the kid's not diagnosed with a retinal detachment at birth, then the visual expectations are good, with the caveats that things like a lazy eye or a need for glasses can kind of delay visual maturation. There's a subset of kids with CTNNB1 that seem to have an optic nerve problem also. That's where the wire that connects the retina to the brain is not functioning normally, and that limits vision. I think we know very little about that. So if the kid has an abnormal optic nerve, then that is going to sort of lower the visual expectations also. But by and large, kids with CTNNB1, unless they have a congenital vision change, should see well. And I think that meetings like this are really going to help us understand more about what our the kids with CTNNB1 are experiencing and how we can help them further. So with that, I just want to thank everyone for inviting me and open up to any questions here or on the Zoom.
Do you have any recommendations on how to talk to maybe your ophthalmologist about getting a referral to a retina specialist? We were fortunate, however, I know there are a lot of parents that have gone multiple times to try to get a referral and have not been successful. So do you have any suggestions for those parents to help them get to the right retina specialist and to the end?
So for those on Zoom, if you couldn't hear that, the question was how do you emphasize to a pediatric ophthalmologist that the kid needs a fever exam with probably a retina specialist? So that's a question I've heard a lot actually. Besides bringing information with you, you could drop my name or send me an email, and I'm happy to help, because I do think it's important. And I think that the awareness in ophthalmology is not uniform. Even for me, I didn't know about this for several years. I mean, I'm just learning more and more about CTNNB1 every single day. And so if you hadn't heard about it before, it's hard to really push for that kind of thing. But I'm happy to be a resource if needed, or you can provide them with information about that.
Or just get a job.
Or yeah. Or,
have seen a pediatric retina specialist who they've done the stage 2 laser
yes.
on our daughter. And now he is saying that fever is the same as retinopathy in preemies, and that now we don't need follow-up care. And it seems to me that he is wrong, and we need to continue to get follow-up angiograms. Is that correct?
It's hard to, first of all, I'm glad that was discovered and treated. That's the most important thing. And then follow-up, it kind of depends on the presentation and the patient. I think at a bare minimum, pediatric ophthalmologist follow-up is necessary. Whether or not a follow-up angiogram is needed is harder to say. I wouldn't say it's a black and white issue, but the most important thing is that the treatment was done.
We had a question on Zoom asking if you've seen patients with droopy eyelids with CTNNB1, if that's indicative of anything.
I see. The question from Zoom was about droopy eyelids, and the name for that is ptosis. And I would say that I haven't seen a lot of that, but just sort of going back to this group of kids who were displayed from the paper here, there are a couple of kids in this group that have droopy eyelids, and so maybe that's something we could learn more about and characterize better in the phenotype going forward.
You mentioned just now the problem with the optic nerve. Could you explain a little bit about how is that diagnosed, or what are the symptoms? What would that symptoms of that be?
Yeah, so this I think we know the least about. But the optic nerve, like I said, is sort of the connection between the retina and the brain. So the retina could be working normally, the brain could be working normally, but the vision's still not good because the connection is interrupted. Usually that's diagnosed if a kid doesn't have visual behavior that you'd expect, meaning they can't navigate a space, so they're just not seeing well as you would think. You can also diagnose it by looking in the eye, generally speaking, and seeing what the optic nerve looks like. Is it smaller than it should be? Does it have the right color? And then sort of the ultimate test often is an MRI to really elucidate if there's a true connection problem based on the size and characteristics of the optic nerve.
So like looking at it through an angiogram, you would be able to see
An angiogram doesn't help too much for that particular
a
question.
regular exam.
Looking at it, usually you can tell.
So I have a question. Our daughter was checked for fever with a dye at the age of 2. It was negative. Would you recommend being rechecked? And if so, what is the difference? You know, how many years would you let that pass before rechecking?
This is a challenging question. So the eye undergoes massive growth between birth and sort of 2 to 3. And so I think if the kids screen kind of less than 3, they probably deserve one more screening. But if it was stone cold normal, when that has to happen, I don't know. I'm a big advocate for combining exams under anesthesia with anything possible. So if the kid needs this, that, and the other thing, if it looks like in a year they're going to need an MRI, then wait, you know, and do it with the MRI. Or if they are going to need some sort of dental procedure, who knows, combine it with that. Just find an opportunity to minimize the anesthesia. But that's my gestalt. I don't know because we don't have enough data yet to say what happens to kids with CTNNB1 fever with time. Any, go ahead.
So you're saying you don't have enough data. So is that something that, well, you might not know the answer to this, that we're capturing with things like this, or are there ways that we can help as a community to get that data to the right people?
Yeah, so it's something I have a strong interest in. And I'm sort of building my network. I was talking to Dr. Chung earlier about how to combine all this information and put it together so that we really know, because all of the data about fever and CTNNB1 is mostly the worst of the worst. And I think there's a lot more that's not that bad, but still vision threatening. And that's harder to capture. And so I'm working on that with your help, largely. And so we'll have more information, just like all the other teams at Phenotype relationships with time.
Just to clarify, we're all concerned about kind of what's going to happen in the future. Is this progressive? Like if it is treated, does it happen again? If it's, you know, after that, used to be 3 years old, you know, get the exam at 5, is that it? Yeah, keep again. And if they were at stage 1 or 2, you need to keep looking, you know what I mean?
Yeah, so the question for Zim was what do we know about the progression? If we think about fever outside of CTNNB1, then usually untreated it will progress. Treated, meaning if it's stage 1 or stage 2 and they have laser, or if it's stage 3 and they have a retinal detachment repair or something like that. Generally speaking, the disease can be controlled, but it requires more close monitoring. So basically, kid with no disease gets very infrequent monitoring. As you have more and more eye disease, you get more and more frequent monitoring, and then you know whether or not another intervention is necessary. So it really depends on the case, and so it's hard for me to speak generally about any one kid. Am I okay for time?
Probably. One more question.
All right.
Thank you so much. Well, I know who I will be making an appointment with to get John's eyes checked now. And with that, I'd like to introduce Dr. Jacob, who actually attended our very first fundraising event in 2019 and has been extremely dedicated to our disorder ever since. So very excited to have her present to you today.
Hi, I want to thank the conference for organizing this great conference. It's really been special to see all these families and see your children and meet with you in person instead of on Zoom. And I'm really happy to have this opportunity to tell you about the work we've been doing to get a deeper understanding of CTNNB1 syndrome and to identify corrective treatments. So I have been studying CTNNB1, the protein encoded by CTNNB1 beta-catenin for, I would say, approximately 16 years. And I really was mostly focused on the brain using mouse models and doing manipulations in neurons of the brain, nerve cells, to do gain of function and loss of function. And I was doing this because I was really interested in the fact that CTNNB1 is known to be a very significant risk factor for the disorder of intellectual disability. And I really wanted to understand more about what it was changing in these nerve cells to give rise to these symptoms. And I had published several papers from the lab on the gain of function and loss of function. And in July of 2019, I got the email that you see here from Alessi Monti, who's a mom of Carlotta in Italy. And she said she spends her evenings looking at the scientific literature, even though she's not a scientist herself. And she found one of my papers on loss of function beta-catenin, and she said, would you help my children? Help all the children in that syndrome. And after several emails back and forth with Alessi, she put me in touch ultimately with Lorraine Cochran and Nicole Vashil. And I went to the first fundraiser that they had in September of 2019, and that sort of started everything for all of us. Because I did not know very much about CTNNB1 syndrome before I had heard from Alessi, even though I'd been studying beta-catenin. And so looking at the literature, as you all know very well, it's a monogenetic disorder. It's caused by sporadic mutations in CTNNB1. Several mutations can occur across this gene. And they're, for the most part, pathogenic. They cause a loss of function, the haploinsufficiency. So you have one healthy gene and one impaired gene. You know the symptoms. The most important thing really we have to focus on is what can we do about treatment. So in looking at what's known, it's really clear that we didn't know enough about the underlying molecular changes, the pathophysiology that were causing the symptoms. And then we didn't know very much at all about any treatment possibilities. And so I got very motivated after I met with the families and decided I would take this on as something my lab would be focusing on. And so we've been working on it for the last 3.5 years. And we set 3 major goals that I outlined here. We decided we're going to generate models. There are not good preclinical models out there. People have not really studied mouse models with CTNNB1 haploinsufficiency past birth. And we really wanted to develop a model for the preclinical in vivo mouse studies, but then also to expand to human cell studies, where we could take the different mutations, some of them from your own children, and really understand what's going on. What are the molecular and functional changes caused by haploinsufficiency or by the different mutations that might have different outcomes slightly, and you'll hear more about that in the next talk. And then we wanted to test drug treatments. DNA-based treatments, because several of the families said, what about gene therapy? So we've started doing that, and a very novel RNA-based therapy. So I think we've made amazing progress in 3.5 years on all 3 goals, and I will share that all with you now. And the follow-up talk is from John Alexander, who's a research assistant professor in the lab, and he'll tell you about our work on the human cell models. I'm going to focus on the mouse in vivo models. So this is kind of the biggest slide. And so this really summarizes everything I'm going to tell you about. So after this, I'll show you some of the data, but this will really give you the take-home message. So I broke it down to what have we accomplished so far. So we developed and characterized a preclinical, what we call an in vivo mouse model of CTNNB1 syndrome. It took us a year to generate this mouse, and that's what we did for the first year. And then we gained novel insights into the molecular and functional changes that occur caused by the haploinsufficiency. And we've identified a small molecule treatment that corrects the phenotypes, the significant improvements in the cognitive, the learning disabilities, and the motor skills in the mouse model. And what's going on actively in the lab now is we're looking at other variants of this small molecule to see if they have better properties. Might they be more stable? Might they get into the brain better? Part of the reason is some of these variants of this molecule have been worked on by pharmaceutical companies, and they may have done some of the preliminary tox studies that we need that are very time-consuming and expensive. So we want to go through these molecule variants and see what is the best one that's a lead compound that we could ultimately get into clinical trials. That's our goal. We have a patent that's pending for the method of treatment for CTNNB1 syndrome. And yesterday, John submitted a paper. It's our first paper on the CTNNB1 studies in the mouse and the treatment. And John and Leanne, who are both here, are the first 2 authors on that paper. We are working with multiple preclinical mouse models human cell models. And John is going to be talking all about the human cell work. We're testing drug treatments, that same drug treatment in the mouse, we're testing it in the human cells. And we're expanding, as I said, to DNA-based and RNA-based treatments in both models. And what we're planning to do is to work with some of the de-identified patient-derived cells that you talked about from Simon. You heard that from Wendy. The cells that are taken, blood cells, are turned into pluripotent stem cells, basically. And those cells can then be differentiated into disease-relevant cell types, like a nerve cell in the brain, or a cell in the spinal cord, or muscle. And so we can look at a series of mutations and really understand them. We have some other human cell models John will describe. We want to conduct tests for safety. You can do a treatment that's effective, but you want to make sure it causes no adverse effects. And we've started a little bit of that, and we have to do more. We haven't tested age of treatment as a variable. We go through one age. I'll show you that data. We want to identify biomarkers so we can monitor the response to any treatment and make sure that everything's working the way we hope. And we're interested in and been attempting and have to continue to engage pharmaceutical companies to work with us. So you've heard a lot about beta-catenin. I'm just going to very briefly summarize what it does. So the protein beta-catenin is expressed in every cell of your body. You cannot live without both copies of the gene that encodes this protein. It's essential. And it has different roles in different cell types and in different stages of development. What we're mostly interested is in its role here, where you see this is a little schematic of one little synapse, a little spine. The synapse is this region of contact between a nerve cell and its target, could be another nerve cell. It's the site of rapid information processing. It's critical for brain function. Beta-catenin is concentrated at that synapse. And what it does is it interacts with adhesion molecules that bring the pre- and postsynaptic cell together. It controls the formation, the maturation of that synapse, so it functions properly, how many synapses you have, the density. And it can change the size of the synapse. So when you undergo learning, the synapse gets larger. Beta-catenin does all of that. And so that's why you have cognitive disabilities. If that function isn't working properly, it's not going to be the same as in a wild type. In addition to functioning at the synapse, beta-catenin accumulates in the cell body and moves into the nucleus with activity. And in the nucleus, it will turn on expression of particular genes. And again, those genes play different roles in different stages of development. It controls things like proliferation, but later in development, it controls things like genes that are important for learning and memory, a plasticity function. And so those are the two most important functions that beta-catenin does that are critical to the symptoms. So we developed the mouse model, as I said, and the idea was to have the whole body have only one copy. Every cell in the body would have one copy of beta-catenin missing, and the gene encoding beta-catenin missing. And so we made this mouse model, and it shows the key features of the symptoms that we're seeing in your children. And so we think this is a really valuable preclinical in vivo model for testing treatments. So the first thing we did is, okay, if beta-catenin is haploinsufficient, we should have a decrease in the messenger RNA that encodes beta-catenin, and we should have a decrease in the beta-catenin protein levels. And you can see we have about a 50% decrease in both. We looked in different parts of the brain, and we also looked in muscle. Then we said, what are the symptoms that the mouse shows? How representative is it? So we did a test for learning and memory in the mouse. And the test that we use is called contextual fear conditioning. Basically, it's associative learning. And the part of the brain called the hippocampus is your learning and memory center. And so associative learning really strongly happens in the hippocampus and the cortex. And you take the mouse and you put it into a novel cage. The cage has never been in. The cage has a wire floor. And it looks in this context, and you give it a very mild flick shot. And it's very unhappy with this situation. So anytime you put it into that cage again, it freezes. And how quickly it learns to freeze shows learning. So the blue is the control, and you can see the control sibling, these are littermates. They freeze very quickly. They're like, you know, this is not where I want to be. The red are the haploinsufficient mouse models, and they feed less. And so we see that there is a learning deficit. So then we wanted to also look at motor skills, and one of the tests was to look for motor sequence learning and motor coordination. So we use what's called a rotor rod. You can see it in the upper corner. It spins very quickly, and the mouse has to coordinate and figure out how to stay on. And it falls off. It doesn't fall far, it doesn't get hurt. So the latency to fall is what you measure. And again, we see a greater latency, less of a latency to fall. The wild types can hold on more, and the het mice don't. We looked at the grip strength, how much force they can generate in muscle, it's reduced. But if we look at just moving around in the open field, total locomotion, speed of movement, they don't seem to be significantly impaired. So we're seeing learning deficits and motor deficits, and we think that's representative of the disorder. So now, if we know molecular changes, we know functional changes, really, how are we going to fix them? That's the critical thing. So the lab really focused in on glycogen synthase kinase, GSK3. As I said, I've been studying beta-catenin for a long time, and it's been really heavily investigated in the field because it's been implicated in certain cancers with gain of function and with reduced function in things like Alzheimer's disease. So there's been great interest, and a lot of different regulators of GSK3 have been investigated. So we did a deep dive into the literature, and we looked for what was the highest specificity small molecule inhibitor of GSK3 that was available. And the idea is GSK3 is constantly active. It's phosphorylating beta-catenin that leads to its degradation, and it's a way to control the levels because you don't want too high or too low. Really important to control. If you inhibit this constantly active enzyme, beta-catenin isn't phosphorylated, so it can accumulate and you can increase your levels. That's the idea, right? First thing we did was to say, okay, let's see if it even does anything to the phenotypes. Does it show corrective responses? That's important, right? So we took 6-week-old mice. We decided we wanted to work with what we call young adults, late adolescents. We didn't want to start very, very young. And we treated them for 5 days or 7 days, 1 injection a day. And we found these results that we think are, to us, incredibly great. This is the contextual fear conditioning. The gray is the drug-treated heterozygous mouse. It's performing at the same level for learning function as its wild-type learning. There's no significant difference between that drug-treated mouse and the wild types. We looked at grip strength, it's significantly improved. It's not quite up to wild type levels. In all cases, it's better than the vehicle-treated heterozygous mouse. We've talked a lot about you want to get beta-catenin levels into that healthy physiological range with any kind of a treatment. You don't want to go too high and you don't want to be too low. It turns out that this drug treatment brings it right up to the healthy physiological range that you see in the matching wild type littermates. It's not too high and not too low. It looks really nice, not significantly different. So we're very excited. Is it safe? We just started doing some pilot studies. We obviously have to do more. We took the wild type littermates and gave them the same exact drug, same concentration, same treatment paradigm. And we see no change in their learning and memory, no change in their muscle grip strength. So that looks promising. The beta-catenin protein levels don't look abnormal. They're not significantly different from the wild type over that same treatment protocol. We have to do more. We have to do longer-term treatments as well to make sure that it stays safe. So one of the other things that we started to do in addition to pursuing the small molecule drug treatment is we were told that a lot of the families are interested in gene therapy. And so we wanted to test the concept, could gene therapy work? Because you hear about this one and done, but if you put this in and you get a protein level expression that's too high, that's not good. And so we just wanted to get some feeling for it. And this is stuff that's relatively new, ongoing. We decided to start with this particular muscle-targeted viral vector. And the reason we started with that is you've heard it's really not easy to get into the brain. This is muscle-specific, and it's been adapted so that it does not go into the liver very much, which would avoid adverse effects. We didn't want any compounds. And it doesn't cause an immune reaction and other adenoviruses that people are using do. And so this seemed to us to be kind of like a really nice proof of concept test. Does this work? Can it work? And how high are beta-catenin levels? So I'm showing you the in vivo mouse work. John will show you some in vitro muscle work, human muscle. Basically, injecting in the mouse one time, and we do see a significant increase in the beta-catenin protein levels, and it's not very high. So we're pretty happy with this, and this is something we're going to be doing more on. So what's the big take home? We've developed some, we've identified novel molecular and functional changes that I think dramatically enhance our understanding of the pathophysiology, give us molecular targets. We know what has to be improved to give you correction of the phenotypes. We have the first proof of concept evidence we're aware of that provides a corrective treatment, and we're really encouraged about this. It's an in vivo small molecule that targets the GSK3 inhibitor. It improved learning and motor capabilities. The mice were treated at a young adult age, which gives us a window of opportunity. So this is after onset of symptoms. So we don't have to worry about getting there really early, which I think is going to be safe and gives us a wider therapeutic window. And I'm happy about that. It normalized the beta-catenin and sodium ATPase levels in the mouse model in vivo. And we so far don't see adverse effects in the wild type. So at this point, I'm going to turn it over to John, and he's going to tell you about the human cell work that we've been doing with the same approaches.
Well, first of all, I'd like to thank all of you guys for having us here so we can talk about this with you. I've been working with Michele for almost 13 years now, and so I'm heavily steeped with the science of this pathway. But I'm not going to belabor the point with more science talk with you in terms of understanding the genetics and the molecular structures and all that. But I do want to point out what our next directions are in translating basically the move from the mouse to the human studies. So we basically created two different lines that basically one is an adult-derived line and one is a fetal-derived line. They're what's called isogenic lines, meaning they have no other known mutations. And what our plan is to do is to create basically the heterozygote version of these human cells in both these lines. And then use the treatments we're working on, including the small molecule inhibitors, as well as the RNA and DNA-based therapies, to see if the results we're seeing in the mouse, not only the pathophysiological results, but whether these treatments can correct some of these things. So basically, the idea is to compare the mouse data and the human data and say, is it the same? So from these inducible pluripotent stem cells with our isogenic lines with just the CTNNB1 haploinsufficiency, our plan is to generate central neurons, spinal cord, spinal motor neurons, and skeletal muscle models. Basically, because those are basically the core pathological cell types that are involved in CTNNB1. So basically, like I said, we have one adult and one fetal derived line. And these, again, lack no other pathogenic mutations. So any changes we see, we can know that are really due to the fact that CTNNB1 is reduced. So we're also partnering, again, like we said, with Simon's Searchlight, basically to get the patient-derived stem cells, and then basically do revertance, where we correct the mutation that's involved and see if it rescues the phenotypes that we observe in these other models. So again, we've all seen the lollipop plot at this point, and I just want to reiterate again that there's mutations that cover the entire sort of beta-catenin gene and covers different functional domains as well, and sort of the interacting partners of beta-catenin. So our idea in terms of working with the patient-derived cells is to kind of get a sampling from each of the different parts of the gene to make sure that we can understand how the mutation affects function and whether we can stratify patients based on where these mutations occur. So one of the things that's kind of unknown is what happens with these mutations. So we believe that a lot of them result in haploinsufficiency or about 50% reduction in beta-catenin levels. But sometimes there's going to be potentially a protein product form. Now, is it a stable product? Does it retain its ability to interact with its binding partners like normal? And if so, is that impact positive? Is it like retaining the normal function that you'd expect with that interaction? Or is it a negative interaction in terms of basically interfering with the wild type version of the beta-catenin protein that's on the other gene, the other allele of beta-catenin, and inhibiting its function? And that's something that's just not known at this point. So basically, again, go sort of looping back. The idea is to characterize these beta-catenin functions in human cells, not only with the isogenic lines where we just delete beta-catenin, but also in the patient-derived lines. And then test the effectiveness of the drug, the RNA-based therapies, and the DNA-based therapies to see if we can correct some of the molecular phenotypes. So, like I said, again, we're going to be generating basically different cell models, and we're going to be generating the ones that we feel are sort of core to the pathology of the disease. And this is a lot of information on this slide, most of which you can ignore. But it's basically showing that you can take these cells and really show at different sort of stages in terms of their differentiation that you're really generating the types of cells that we're actually interested in. So to sort of get into actually now sort of some of the data that we're actually about to see and some of the exciting stuff that is in parallel what we've been seeing in Vivo. So in collaboration with a lab who does a lot of stuff in terms of ALS and who actually does myocytes, basically muscle cells a whole lot of times, we actually got a couple sample wells in a very quick pilot study with her and just did some cellular lysates to see, number one, can detect beta-catenin and also maybe one of its functional sort of downstream targets. So in these human iPSC-derived myotubes, we're actually able to detect beta-catenin and also one of its Wnt target genes, axin-2. Now, Michele did talk about how we use this myo-AAV, which is a muscle-specific AAV virus that will actually expresses the beta-catenin gene. When we actually put these in these human-derived myotubes, we actually see, number one, increases in beta-catenin and also an increase in the expected level, in the expected Wnt target gene, which is axin-2. So this is again, this is a very small n. This is n of 1. So, but it's very encouraging in terms of, number one, that the AAV is doing its job, and then we're hitting the right cell type. We are currently in the process of differentiating all of these human-derived lines into, first of all, cortical glutamatergic neurons, basically the neurons of the brain, and a couple upcoming experiments in the next few weeks and months is going to be testing, number one, the small molecule inhibitor, and also one of the RNA-based therapies to see basically if this thing that has actually been shown to be able to actually function in the mouse brain, whether we can increase beta-catenin in the human cells, and especially human neurons. So sort of the general takeaways from this is we've created this new mouse line that does show reduced beta-catenin levels. And this model will help define some of the pathophysiological effects of reduced beta-catenin that we may observe in the mouse model and help us also define possibly new ones that actually are involved in the humans. So we actually have new pilot studies that show that gene therapy can increase beta-catenin and Wnt target gene expression. And we're also providing the first proof of concept of the effectiveness of the drug for treatments to significantly improve beta-cat levels in human cells. The big bottom line is the treatment of this small molecule inhibitor is highly specific for GSK and is providing promising results in two different preclinical models, the mouse and the human cells. So on that note, I think we'll keep it quick, and Michele and I will answer your questions.
Had one question from Zoom, asking what is the targeted timeline for completion of the different trials in human cells?
Well, so as far as the haploinsufficient lines, this is something we're very new at as a lab. So it's going to be number one, differentiating them to the various cell types, and that does take time. And number two, then being able to test all the different treatment modalities. So as a specific timeline, it's going to be very hard to pin down, but we're working hard on it right now. And we just got these cell lines about a month and a half ago. And so these are all very, very preliminary pilot studies.
Got another one online as well, just asking, kind of in your opinion, what assets, tools, or studies might we be lacking currently, and where should we be kind of focusing?
I think right now, I can't think of anything we're absolutely lacking other than probably money, people, time.
That's the next question.
I was going to say.
John spent the last 21 days in the lab every day, so he wants more people. I think we have the ideas. I think we have what we need. What we really need is, I would love to get some kind of major backing, some angel investors and pharmaceutical company that would help us do this so that we could get, all the toxicology studies that we need and really make progress on what we've accomplished so far. Because I feel like we have the beginnings of something really important and we just need to keep going with it.
As someone who, I have no idea how much money that would be, like how much are you, what would you ask for from a big donor?
From the research or to move it to a clinical trial?
So clinical trial with the medicine.
What I've been quoted, because I'm not expert at this, but I've talked to a lot of people who have done this in their lives, Gerald, Cobb, Wendy, Chung, and other people from major pharmaceutical companies. Toxicology studies can cost, them alone can cost about $1 million. And then when you go past the toxicology studies to starting the clinical trials, it's a couple of million.
At that point in time, it's usually a pharmaceutical or biotech.
We are trying to engage pharmaceutical companies, large pharmaceutical companies that would be interested because some of them have worked with some of these molecules because of looking at it for Alzheimer's.
Yeah, I think Michele's approached, 5 different pharmaceutical companies in the last 2 months. So
Do the human, what do you test first? When we're testing in humans, do you test from blood products or do you test an actual human, like what do you test for mice to like? I'm just really curious about that.
The blood samples that your families are donating to Siedman's, those blood cells, there are cells that are taken from there, white blood cells that are then programmed to de-differentiate, to become pluripotent. And the value then is that they can be then differentiated to any cell type. And so Siedman's is taking them and generating, Wendy talked about it, what we call these human-induced pluripotent stem cells. They can be turned into what we're going to do, which is brain excitatory neurons. We could also do inhibitory neurons. We could do glia, but we're going to start with the excitatory neurons, spinal motor neurons, and muscle cells. And so you can differentiate them to these different cell types and then test the different treatment therapies. So we have selected, I think we're starting with about 6, and we might add a few more where we took de-identified mutations from the Siedman's collection that look like they're going across the whole gene. They're different mutation types because we really want to know, does the treatment work? Which of the different treatments works best, and are they safe? We want to make sure there are no adverse effects. There might be certain mutations where there's a partial product we don't want to increase the level of that partial product, and if one of these therapies increases that and it doesn't improve the phenotypes, that would be advised then to go ahead with that treatment with someone with that mutation. So we really are trying to be very cautious and very thorough.
You mentioned you have a paper coming out, I think you said tomorrow.
We
No,
submitted
we
it yesterday.
It hasn't been accepted. It's just been.
We're good with publishing it tomorrow day.
Yeah, I know.
No, I was just wondering, when do you expect to know about that, when the peer review process, and can you tell us what the name of the paper is so that we can be looking for it?
We'll share the paper when the name, it's basically typically it takes 1 to 2 months to get your reviews back. It's rare to get it with no revisions, which basically usually means a few more experiments. So these days, we all consider a paper to require about a year to actually be in press, but you know, maybe we'll be lucky. It's happened once.
Just a follow-up question to this, is there going to be a preprint available on BioRx or something?
As soon as it's in press, we'll make it available to you guys.
Michele, there's another question on Zoom, which I know we discussed about lithium, which I know you studied for a different disorder, and why are we not looking to that further?
So lithium is one of these interesting drugs. It increases beta-catenin levels, and partly it does it through GSK3. But lithium is what we in the field would call a dirty drug. It's much less specific than the things we're working with. It has a lot of other targets. GSK3 has other substrates besides beta-catenin. I don't want to say that it's specific only to that, but we're taking one that has such high specificity. It's been tested against 350 other human kinases, which is the best specificity out there. It's really a very nice molecule. Lithium, on the other hand, doesn't only affect GSK, it has other things that it affects. And it's been in use for bipolar disorder for decades. People can be on it really long term. So in that case, you might consider it relatively safe, but it does cause thyroid potential for thyroid and kidney problems, and it is not very well tolerated in children. And so we wanted to do, in the beginning, we thought we would test lithium, but we really wanted to do something that we felt was much safer and would be more comfortable with.
You mentioned gene therapy briefly. I guess I'm just thinking about, let's say in the future, drug therapy becomes available, and then you drop it that gene therapy becomes available. And you kind of mentioned it's possible. Could there be any conflict, or like, if you did the drug therapy, would that disqualify you with gene therapy?
As far as a clinical trial, I mean, again, we're not experts on clinical trials, but the nice thing about the small molecules Jerry mentioned is that you can always stop it. At least this particular compound itself has a relatively short half-life. So in fact, we showed you the behavioral data in the mouse and how it actually rescued behaviors with the 24-hour probe trial from fear conditioning. However, when we look longer term out, it's back down to the levels of the normal heterozygous mouse. So it's not a long lasting effect. It's at this point, it's something you would have to be considering taking on a possibly daily basis.
If a lot of the children here are relatively young, so looking far out, and then if any of these therapies come to fruition, and these kids are now 17, 18, 20, will they work? Will they give them back all those years that they lost, or by that time, is it something that that's just how their bodies will be?
It's a good question. It's honestly at this point, we just don't know. And what we've seen, at least in the mouse, is we treated these mice at what we call young adult stage for mice. And it did have a corrective effect. Whether it would be better starting younger, and that could also have its own potential for problems itself. We just don't know at this point, and it's something we're obviously looking to study.
I'm kind of optimistic that we have a pretty good window for treating at older ages, partly because of the results of the mouse and the fact that the beta-catenin function and the sodium potassium ATPase function, those are activity dependent, constantly changing, really important for plasticity. And that's relevant throughout all of life. And studies have been done like in aged mice and little pilots in humans where you increase beta-catenin in an aged mouse that has started to show some kind of decline or an Alzheimer's model, it improves learning, even in very aged animals. And so that's something, will it go to completely normal? I can tell you, I would expect statistically significant improvements.
And like on a sort of separate point to that also, it's generally thought of that the younger the intervention, the better. This is something we're obviously going to have to test. But like you mentioned, a lot of your kids are quite young, but you're also probably doing the right things now that you know, like physical therapy, basically helping them out. Those kind of things make huge improvements in their life, and obviously, like, keep it up. You guys are doing a great job.
Yeah, activity increases beta-catenin levels. Keep them active. Don't wear them out. Don't let them wear you out. Keep them moving.
Yep.
Mine wasn't really a question, so I just before you had left, I just wanted to thank you for your time in the lab. Like you said, you were there the last couple of days, and tried to spend some time to interview. So I just want to thank you for everything that you guys have done for.
You guys inspire us and keep us going. And every time we sort of, hit some wall, we're like, okay, we gotta just do this anyway. Keep going. It's been great. So it's a shared goal.
One more question that came in on Zoom, and I think we don't know the answer to this yet, is how are you thinking this could potentially be delivered? Would it be injection, pill form, or is that something as we get further down the line?
We're hoping to modify it to be oral. Right now it's injection, but that's one of our goals is to make it oral because it would be just more readily administered that way. So that's one of the things we're going to move to.
I just wanted to, the point that you made quickly, and thanks to everybody glossed over. You said activity proves baked in. Is there any indication in beta-catenin over the years that there's any natural supplemental dietary things that increase beta-catenin levels?
Not really. I think, obviously good health, but yeah, physical activity. It gets into the sort of the details of the science. We talked about this Wnt pathway. Wnt is a soluble. Going to be a scientist, but I think you can follow what I'm talking about. Wnt is a soluble molecule released with activity. And it binds to these receptors on the cell, and it naturally inhibits GSK3. That's how we came to thinking about GSK3, because you've got this endogenous molecule that's always regulating beta-catenin levels. There are other molecules in the cell that also do it, but Wnt is activity dependent. And so, learning, memory, stimulation, physical activity, all of that, that's how we all improve our cognitive function and our skill set. And so part of that is just naturally increasing the beta-catenin. And I see changes. Like I was noting when I went to the first fundraiser, I met Declan. That was summer of 2019. I see him from then to now. It's beautiful to see how much better he's talking, walking, interacting. So I'm very optimistic that it's keeping your kids active and hopefully getting a treatment in place. Things can be really great for all of the children.
Actually, on a personal note, my niece has a disease called Rett syndrome. It's actually very, very similar in some ways to CTNNB1 syndrome. And obviously my sister and her husband have been great advocates for her. And the physical therapy, skiing, horseback riding, it's improved her so much. So keep it up.
I just first want to start off by saying what an incredible honor it is to be here, and just it's an honor to be here with your families and with your community. And by community, I mean, the scientific community, the medical community, the caregiver community, all the kiddos out there, and learning about the people who are, adults and older, and how exciting that is. And I've learned so much today. So thanks for having me, everybody. I'm Luke Rosen, and I hate two things: bios and PowerPoint. So maybe you'll just figure out who I am as we go along. But the one part of my bio that I think is relevant here is that I'm Susanna's father. And that's Susanna right there. That's our girl. And I was fortunate enough to be asked to talk about our path to treatment, which is constantly evolving. So I'm just going to talk a little bit about Susanna and a little bit about what we're doing now, and also just quickly touch on our foundation, because I always feel as if I'm also fortunate enough to kind of be the voice for people who are in our community, who don't have the fortune to have a voice here on a platform here. So anyway, this is my daughter Susanna, who is now 9 years old. And I'll just give a quick timeline. So Susanna was born in 2014, and it was she was our second kid. And so and forgive me if any of you hear me repeating things that I've said before. Sometimes I do that, but how with your first kid, if there's a bee sting, you're in an emergency room, but with the second kid, you're like, she'll catch up. She's gonna be fine. So Susanna was born, and I was an actor for 20 somewhat years in New York, and she would come to work with me, and her first job was in this print ad that was on buses and big jumbo screens and everything. So it was kind of life. It was great. Susanna would come to me to work, and then she everything was going okay. I'm not gonna go through our diagnostic process, because I feel as if it's probably so similar to so many of you in here, and I don't want to waste time, but I'll touch on just a little bit about it. In 2016, we found out, and I know from talking to many of you that it was a very similar experience. We found out through whole genome sequencing, or whole exome sequencing, excuse me, her genome came later, I'll get there, that she has a mutation in her KIF1A gene. And when we were told this, we were told that there are maybe 12 or 15 people in the world that they find in literature, right? And we immediately kind of knew that that couldn't be true. My wife, Sally, was, I'm glad she's not here because she doesn't like when I say this. She's over 40 when she had Susanna, which is called a geriatric pregnancy. The hospital actually say that. So you have this genetic testing panel, if that's the case, right? And we didn't know anything about genetic testing. So when they said genetic testing, we said, no, we already had that. We're good. We figured it out. We had that. But so we had more extensive genetic testing and found out that KIF1A was the problem, and that there was no treatment and no cure. And we thought, well, how can that be? And what can we do to fix that? And I remember the exact moment I was on set for a TV show called Orange is the New Black. And I was in my trailer and got a phone call from Sally, is my wife, who said, we're going in to see Dr. Chung, who I think just left tomorrow at 7 a.m. And so that is when our family started to grow, right? So here's Susanna when she was 2, right, at diagnosis with Dr. Chung, and when it was, I call it the elevator, that elevator ride from, it's like our elevator ride, I'll never forget, right? We got up there, and the elevator doors opened, and there was a genetic counselor, a social worker, a nurse practitioner, and it was like 7 in the morning. And when we got up there, Sal and I looked at each other and we said, shit. You know, what are we about to do? And so, but, you know, the good news was that our family grew because we have this extended family now, and it's our physicians, our neurologist who's here, who's just, you know, our extended family. And we talk to our doctors and researchers weekly, if not daily. And it's quite a task they have, because it's not just caring for our children, but it's educating us and making it colloquial. So I had to, you know, so we can really play a part in conversation. And that was something that took some time, but we kind of got it quickly. In 2017, we had started the foundation, and this was Susanna's world. She was just not present. She wasn't. There were times where she would just go away, and we would have to get her back, and that was hard to do. And as we found more and more people, up in the right corner, you'll see red names. We started finding more and more people. I think at this point we had 45 people, and we started a registry, National History. But our registry wasn't just growing. You know, this year in 2017, of the people we found, Gustav and Braden had died. And that was a hard pill to swallow as we became really, really close. As you know, our communities are, you know, this is our tribe. And so Susanna's world was not quite present, that's how I would say it. And she was having, we found that she was having seizures at night that were non-convulsive, but they were, you know, just as catastrophic as another seizure. So that was really playing a factor in her. In 2019, she stopped being able to really walk, or to certainly she couldn't get up unassisted, because she was in so much pain. She has some peripheral neuropathy, and she's very spastic, and she would just start falling, breaking bones and putting her teeth through her lip. And yeah, that's when we decided that it was time to really, you know, it's time for a wheelchair. And so the last time she stood without assistance, you know, we could pick her up, we could get her up and hold her hand, and she could grab onto something and pull up, but she couldn't stand up. I know a lot of your families face that same challenge, so you know where I'm coming from. And that was a hard realization, right? When you realize that, you know, we have these diagnoses, but when you realize that, holy shit, my kid is gonna be in a wheelchair. And I remember so specifically when we finally got medicated to send the wheelchair, which took way too long, building it. Like I was screwing it and putting it together. And I was so happy that we finally got this wheelchair. And I called my brother and I was like, you know, I can't believe that I'm happy that I'm putting my daughter's wheelchair together. So that was in 2019. I'm going to cut ahead. Like 2020 was more of the same, right? I'm not gonna. You guys have all been there. So 2020 was more of the same. But what had happened in 2019 and 2020 is there was a program called NMORA, right? And it had just started. And they were just starting to accept a few applications. And when we applied, and we got the news in late 2020 that Susanna had been accepted and was going to be likely the first person to be dosed. So we were like, wait a minute, what? Okay, we've spent all this time with our foundation and funding research and you know, lemonade stands, a very blue collars, you know, foundation. And but now all of a sudden it's like, you know, it all, when we were accepted to when Susanna was first dosed, felt like a second, but it was 3 years, it's 2 years, you know, 2.5 years. And so I also think, as I've met many of your kids who are unaffected, is that I could very, very clearly say that our life has changed because of our son, Nat. And I meet siblings like Esme, and I meet all of your siblings, and and they're just remarkable. And it's something that is more than any drug or any therapy. It's the responsibility that our children have. So what happened next is it was in the middle of COVID, and we had what is called a long read sequencing, which is a very more extensive genetic test. And that was hard to fight for to get because Illumina's, excuse me if anyone here is from Illumina, but Illumina's machine broke. And then wait a minute, I've been to Illumina. There's not one machine there. You know, what is happening here? And so we had to kind of, we had to kind of fight to get another company to have the kindness of their heart to send someone over. So Suzanne was getting her genome sequenced again, only with greater detail to find the polymorphisms that many of the scientists have spoken about here. And there's my son Matt holding her hand, and my daughter or my wife. Nat is with Susanna all the time. And cut to there was a lot happening before the actual dose day. There were baseline visits. There were toxicology studies and FDA meetings, and all of these things that were terrifying. There was not much safety known. There was not, Susanna was the first real person to get this ASO, certainly with this gene. And so forgot to mention it's antisense oligonucleotide, or an ASO, which is the treatment that Susanna is on. And the first day where we got our day where she was being dosed, this picture over here is we had to be in the hospital pretty early. So this is wake up. Nat gave her a big hug goodbye. And I can only imagine what was going through his mind, because he was old enough to Google things. He's old enough to know that a week before this, there were, and my heart goes out to these families, that a week before this, there was a child who died from the same administration and the same sort of drug, albeit a different disease, and it's very terrifying. But we didn't have many other chances. She was rapidly declining, and anyway, this was treatment day. And so we woke up, we went to the hospital. Susanna was pretty scared. I mean, Susanna is the kind of kid who, she loves the hospital. She calls, the food that comes in her room, she calls room service. She, she goes to get her blood test. She just does that. She loves the hospital, but she can sense that something's happening here. And then our extended family, and I'm gonna cry at some point, by the way, the next 10 minutes or however long I have left. Jen Bain, who's to the right over here, who's Susanna's neurologist, and who is somebody this involves so much trust, that we trust you so emphatically, because this is somebody who puts an intrathecal lumbar injection into our daughter's body with a drug that has never been used for this disease before. And Jen, or Dr. Bain, has quite literally changed our lives, because a few days later, after our observation, Susanna was up and holding on to Jen, and they were giving each other a hug, and we could go home. And we live close to Columbia, so that was a little bit of solace. Like, if anything goes wrong, we can go right to Columbia, where we have gone to the emergency room several times for multiple different things, and it's always Dr. Bain who's there. Even if she wasn't there, she would beat us to the hospital from wherever she lives. She's right around here. There was never one moment where, and this is when I talk about, you find your community, and it's the scientific community, the medical community, and our community, the patient community, is all one community. And you find these people who love your child so much. And that's Dr. Bain, that's Dr. Chung, that's myriad of people we've met. And it transcends our genetic community, right? I think our community is gene agnostic, right? I listen to Effie's podcast And I learned so much about other people who have challenges that we do, but are in the same situation and answer questions that we have. So I think that we're due to grow beyond just our gene. And that's what I hope happens really soon. And then all of a sudden, we saw change in Susanna. This is a picture of Susanna. If you remember, I said she stopped standing up on her own. She lost a lot of skills, but she stopped standing up on her own when she was in 2019. So this was after, I think, dose 2, October, dose 2. And she stood up on her own. When we have the video, I'm going to show you the video because the file was too big. But I was, of course, weeping behind the camera, as Sally was very composed, and Susanna was explaining to Sally that this is how I'm getting up. So the words were starting to. She always had these words on the tip of her tongue that were starting to come out. And I don't know if it's the drug. She also has an incredible speech therapist, an incredible physical therapist. Maybe it's everything together. And however it's happening, we start to see change. And during this time, so this is the first time she stood up in 4 years. And during this time, my father was very sick. And the next day, I showed my father this video of Susanna standing up for the first time. And my dad died 2 days later. And I really, in my heart, believe that my father stayed alive to see that Susanna was in the hands of people like Dr. Bain, in the hands of people who were committed to making this drug for her, and that there was progress happening. And yeah, sorry. Okay. He had this, he was a doctor, he had this this quote on his door that forever, since I could remember. And it's a George Bernard Shaw quote from this incredible play called Man and Superman. Some of you might know it. George Bernard Shaw was not very good with nouns and pronouns, so excuse his, just the word man. But the quote is, "The reasonable man adapts himself to the world. The unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man." So if you think of that, like, this is a room full of completely unreasonable people right now, because we're doing this. I mean, do you ever think that we would all be in a room like forging research and for our children to have this disease that just has letters next to it, and so you have to not just think outside the box, but completely get rid of the box. And I feel like, and I'm not a scientist and I'm not a doctor, but I feel like there's something very creative to what they do. And it's a way of pivoting when you need to pivot, and it's a way of looking at every person as a person and not as a cell line or a phenotype. It's a person, and every person is different, and that's why we're so lucky to have so many people in our corner. That was on Dad's door. And then I saw more change. So I hope you guys liked all the rigs that came over. But so I'm a firefighter, and that helmet weighs 8 pounds. Susanna has a very weak neck, and we're not sure if it's vision, we're not sure what's going on, but she has a severe tilt to one side of her head. And for the longest time, I thought her neck just wasn't that strong. And then I looked over, and she grabbed my helmet and put it on. I was like, that is 8 pounds in this girl's head, and she can hold it up. That's not a standardized FDA approved measure by any chance, but it still, for me, was a moment where I thought she's getting stronger. And I'm going to jump back to 2017 just because I want to talk a little bit about our organization. And we started a And Dr. Bain spoke at it. We started to find more people, to more unreasonable people. And we started to find this community that kept growing. And the first family meeting was about 30 people. Some of them were on phones, some of them were here. It was at Columbia, was kind enough to host it. Dr. Bain came in to talk about some of the neurological things that were happening. There's an ophthalmologist that came in who talked about the eyes. There was people coming in to talk about the spasticity. And the answer all the time was, he told us everything about the anatomy and the biology, but the answer was, we don't know. Right? No one knew. So it was, it was this turning point. We go one way or the other. We could say, okay, no one knows, that sucks. Or we could say, no one knows, let's go figure it out. And thank goodness we have such a strong community that it was the latter. And then we started finding more people. We started finding more kids. And our community exists, and if our KIF1A kids were here with your families, this would happen too. It's not because they share the same gene, it's because our kids are so special in so many different ways. And it's not special needs that we fight for in our IEP to get certain services, but it's this inherent something, I can't name it, that's in their heart. And I don't know what it is, but these kids hold each other up. And when we put them together, something incredible happens, smiles happen, and people look healthier, and then up here you'll see Eleonora and Ryan. Eleonora is somebody who's amazingly close to my heart. She is in the middle where Sally is holding Eleonora. They're a family from Sweden, and they came over to New York. Sally was singing to Eleonora, was singing Wheels on the Bus. And Ricardo, who's become a great friend of mine, who's Eleanor's father, said, that's the first time that she's reacted with happiness. I mean, Eleonora was severely affected, and she had this little smile. And Susanna came over and put her hand on Eleanor's head, and Eleanor died 3 days later. And it was the first time where this future was right in front of us, and it was, what are we going to do? And so thank God for science, and thank God for medicine, because we're working really hard. If you saw Dr. Chung's slides earlier and you saw all of those assessments that she put up, those were Susanna, that was Susanna's schedule. It's grueling. It's really, really, being in a clinical trial is hard. And especially one that has no precedence. But then there's nothing can happen without hope. I would talk to Effie about hope as like a baseball player's swing, right? Like I coach my kids Little League, and every time he gets up, there's a little, I can say to him, he's in a slump, and I say, you just need to adjust your swing, just like a little bit. And he starts to hit. And I feel like hope for all of us, sometimes at the end of the day, it's just like, how can I have any more hope? I'm so drained. And then I lean on somebody in our community, whether it's a doctor or a friend or somebody, and they just fix my swing a little bit, and my hope comes back. And I think that is a responsibility that we all have, is to fix each other's hope swings. And then to realize that community is everything, and not just our gene community, not just our rare disease community, but our community, it's where we live. Susanna was in the hospital for Christmas 2 years ago, around Christmas. And it was the day that we had our firehouse Christmas party, where all the kids come in and sit on Santa's lap and everything. And we got home from the hospital, and Susanna had missed that. So she didn't get her present, she didn't get to sit on Santa's lap. And we got home, and I went to work. I went to the firehouse, and I have nighttime shifts, so I can be with Susanna during the day. And the alarm goes off, and I thought we were going to fire. And we jump in the rig, and our operator, who drives the truck is dressed like Santa Claus. And we drove, this is our house over here, we drove right to our house. And we told Susanna that we drove up to the North Pole. Got Santa Claus. Took her down to our house for her in a big red truck. And she was so happy because she didn't miss Christmas with the rest of her peers. And it just meant the world to us. And it's family that if we tell our story, if we tell our story, then the whole community gets behind you. It doesn't matter if they don't know what a gene is. If you tell your story and ask for help, you get it. I asked for help a lot. Oh yeah, and then 2020 to 2022, life happened and I got late stage cancer. That sucks, right? And the only thing I can think of, and I've spoken to Effie about the only person that I trusted to, I didn't talk about it much because I wanted to keep the focus on Susanna and her disease and other kids like her. So I didn't really talk much about my cancer. And then one day I was talking to Effie about how terrifying it is what happens when we're gone. And then Effie said, or I asked her if I could come on the podcast and talk about it. And that's the first time I ever talked about it. But that's, I mean, for the time being. And, it was really Susanna that kind of, all of a sudden, there was this empathy that we had for each other. I had peripheral neuropathy in my hands from the chemo. She had, it was something that we just shared. But you don't let it stop you, right? You never, ever, ever give up when it comes to things like this. And so we were, I was doing Zooms from the chemo suite and all this stuff, but also realized people say, hey, it's a marathon, not a sprint. It's going to take bullshit, right? Bullshit. It is a sprint, not a marathon. We are all here because we are trying to get treatments to our kids faster. It's not a marathon, and we can do everything we can to put our shoulders behind the scientific community and the medical community, everything we can, whether it's resources, whether it's participation, whatever it is, that's us sprinting. Having a conference like that is us sprinting. And so here is Susanna this is her when she was having a lot of seizures at night, and we had no clue what was going on. We were terrified. This is when I told you that moment where we decided that she had to be in a wheelchair because she was breaking too many bones. This is after the baseball. Baseball is after the third dose of her ASO. So I just put this one in because I really love these pictures. And this is something that you have to ask Effie about. So everybody after this moment or whatever, this room when you're outside, ask Effie about the marble jar, because when I learned about the marble jar from talking to Effie, it changed my life. So listen, I would love to talk to all of you, and I don't want to take up more time, but again, it's a real honor to be here. And thank you to our neurologists for always being there, and to science for helping us get one step closer to making sure that Susanna is a pioneer for, not just treatment for her, but for, the watershed that comes after her. So thanks for listening. Thanks for having me.
Only you can make a presentation about someone else, and it's so much you, because you're just the most genuine and thoughtful and real person. I've been so lucky being so immersed in the rare disease community to find these rare gems, and Luke is one of them, and our next speaker is another one. These people who are not just doing it for themselves, and they're doing it for the kids that come after. Like Luke mentioned, all that data was Susanna, he's taking this huge risk, their whole family is, not just for Susanna, but for all of us. And I've seen Susanna go from regular sick, hospital kid who's doing things, to doing nothing, to going blind, to being in the hospital all the time, to then seeing a picture of her painting her dad's nails after she got the treatment. So thank you, Luke, for taking that risk for all of us, and I'm so happy to see Susanna doing so well now. One of the things that was really concerning for me after I had Ford was I knew I wanted to have more kids. And so I got really hyper sort of sensitive to like, how do I not screw this up? Because this is going to be really hard for Ezzy. And so I started looking for siblings. And then I found this family, the Siedmans. It's not just Jennifer who you're going to meet, but her entire family plays like this super important role, because I think too, as new parents, we're always trying to look beyond and like figure out what our future is going to look like for our siblings, for our partners, and for our sick kids. And so finding parents who are further along in the journey was something that was really important to me. And I found one of the best ones out there in the world. And she'll tell you about her family story, but what's even more special about Jen is that she's, I don't even know how many years into this now, a couple decades, and she's still getting up every day and offering resources and advice for families. And she is just such a special person, and she has so much to offer families. She's the community engagement coordinator at Courageous Parents Network. This is a resource I send to every new family. They offer videos and all the things like across the board for families who are newly diagnosed, and we have seriously sick kids, and they're just doing an amazing job. So I know you're gonna love everything about Jennifer, just like I do.
So I'm gonna just tell you that I'm a little nervous, even though I speak a lot. I usually am talking about topics like palliative care or goals of care, or like we're gonna talk about coping for the caregiver later on. And when I was speaking with Lauren and Effie, they said, can you try to do something different? And plus I have to come after Luke, who was a professional actor. So I do not have my entire talk memorized today. So if you see me looking down, I apologize, but it's just not in my skill set. So anyway, yeah, this is my son, Ben. You're gonna meet him a little bit through my talk. That was him shortly after his diagnosis. Ben's Dream was the name of our foundation that we started when he was about 5. Proud to say that gene therapy that we started funding, this was 1997. I'm a long hauler in rare disease. Has made it into a human clinical trial. Sadly, it's hit a few bumps in the road, as we can expect often with science, but it is possible. You just gotta stick with it. And this is my husband, Stuart, whose name I never say right. He says that I say it funny, but and I were interviewed by Courageous Parents Network, which is an online resource for parents caring for children with life-limiting illnesses. We can talk more about that later. We were interviewed. That's our platform, is mostly videos and mostly talking about how palliative care made a big significant change in our lives. But anyway, I'm going to get started now with what I really actually wrote and prepared, and hopefully it works. And hopefully I don't mess up the computer. So anyway, Effie knows this about me. She's the one that asked me that I totally love analogies and metaphors. Like I can talk to Effie and I'll always have something. It's probably a little bit like her marble jar. But a favorite of mine even made her podcast. I was really happy about this. It was called The Supermarket Answer. That's the response I would often give when someone asks, Ben. And in that instance, you stand there and you wonder, how am I going to say now? Does this person want the real deal? Or do they just want the supermarket answer? I'm fine, he's fine, it's all fine. Right? You know, and you think about that when you're standing there, right? It's the truth, right? But what you really want to say to yourself is, does this person want my true deal, which would come out in my life like, well, actually he fell yesterday, the day before, the week before that, it was 2 eggs on his head and a trip to the ER to get stitches. So now I'm trying to think about patting my walls. You know, I'm sure you can all relate, questioning to what answer to give and questioning what response it might elicit from the person standing across from you. So now you have a new way to think about it, okay? Remember when the person asks you, oh shit, am I gonna give out the supermarket answer now? I've used metaphors of garbage and a garbage truck to explain the science of Sanfilippo syndrome and sword fights and fire alarms to give others insights into what life with Ben looked and felt like. I think I do this with non-rare audiences in an attempt to make my life somehow more relatable. I think we all do that, right? We want to somehow find a way to relate to other people. But I know when I use these kind of comparisons in the comforting presence of other people, other rare parents like yourself, and it's been really great to be here today, and especially great for me to spend time with the kids because I miss that. It's with the hope that something I've learned caring for Ben and through my work at Creative Spirits Network will somehow be useful to you. So I hope that when you walk away today, you feel like you got some use out of this. And I just want to warn you, there's a lot of analogies and metaphors coming your way. My older son, Noah, and I often walk the dog together. During one of his walks, he told me about a video he watched on sunglasses. Yes, he's 29. He's watching videos on sunglasses on YouTube. And the blocking of photons and Bell's theorem. Yeah, he's a nerd, all right. I'm sure I will not get this entirely right because I am not a scientist. I am not a nerd. But he explained that sunglasses block some, but not all photons of light due to the relationship between the orientation of the lens and the oncoming photons. He told me that lenses layered in a perpendicular orientation will fully block light. But he went on to explain that if you added a third lens to the stack, the light would come through. Bell's theorem, if I understood him correctly. A few days later, I rewatched the movie National Treasure. And between my conversation with Noah and the rewatching, a thought started to form in this crazy head that being a resilient rare disease parent requires having your own pair of Ben Franklin glasses. Now, I'm probably dating myself with this reference to this movie. I'm hoping that some of you in the audience have at least watched it, but just in case, those glasses are an optical tool with rotating lenses invented by, of course, none other than Ben Franklin. And Nicolas Cage's character in the movie uses them to find and read hidden messages on the Declaration of Independence, clues that eventually lead him to discover the Templar treasure, the greatest treasure ever discovered. When Ben was diagnosed, I felt like so much of the light was filtered out of my life. This quote by Emily Rath Black really captures how I felt. There seemed to be so many joys and opportunities and choices that I felt robbed of. So many he might nevers. I didn't know then that what I was feeling was anticipatory grief, the sadness we experience when our life that the life we anticipated for ourselves takes a sudden turn. But I think even in those early days, if I had had a name for it, it would not have stopped me from feeling blinded by them. I was, as in Noah's explanation of Bell's theorem, wearing my sunglasses in a perpendicular fashion, and no light was getting through. With this new lens fixed in place and dashed expectations all around me, it was as if all hope was also being filtered out. This is a picture of Ben, sitting up kid, shortly after his diagnosis, and the day I believe I started to grow my own or build my own pair of Ben Franklin glasses. It was a simple summer day. The boys were playing with my best friend's daughter in a kiddie pool. The weeks preceding this day, I spent countless hours focusing on all the sibling experiences my boys would never have. I even organized some exceptional opportunities, like taking them, look at how old they are, to meet the Boston Red Sox mascot, like they were going to know what we were doing. And it was a stupid attempt on my part to create memories that might never, trying to replace the ones they might never have. But here they were on this great summer day, creating their own memories, building their own foundation, expecting only a fresh supply of water from the hose and hoping only to douse one another with a bucket full. And for reasons I wish I could more clearly identify, because I can't, my lenses lined up and the first important clue in my life was revealed. That expectations and hopes, well linked, are better understood when we see them from two different lenses. And that even as I might always have one lens fixed in place, filtering out some of the light and altering my expectations, I had the power to redefine and refine my hopes. I might no longer be able to expect that Ben and Noah would race their bikes down the street. but I could hope that a feeling of exhilaration became part of their brotherly bond. Years later, I would watch Noah terrify shoppers, zooming Ben's wheelchair at breakneck speeds around the supermarket. It was his favorite thing to do. Short of going, I'll tell you the other story about their favorite thing later. Noah had a grin as wide as the Grand Canyon on his face, and Ben, who could laugh like a mean man, was howling like a hyena. Both of them had cheeks flushed with exhilaration. My hope had come true. Refinding and redefining hopes are a theme we hear often at Courageous Parents Network. In fact, it led us to conduct some research into the family journey to determine what supports are needed at different inflection points. The research revealed that families caring for medically complex children are in a continuous cycle of orienting, navigating, and moving forward. I think the key word there is that it's continuous. So you're repeating it. Often coinciding with the onset of a new problem with our children, orienting is typically the most emotionally difficult part of the cycle. I think this is because these are the times when our hopes feel elusive, and using our Ben Franklin glasses and honing our ability to shift our lenses and change our perspective doesn't come all at once. For those of you who've seen the movie, you know that Nicolas Cage had to experiment with the combination of lenses at each new location before a new clue was revealed. Navigating, it turns out, is typically the part of the cycle where families spend the most time. You guys all know this. Navigating is when you problem solve, and Lord knows we have a lot of problems to solve, right? And I think this is when the colored lenses of our Ben Franklin glasses really come into play. But to test my theory, I had to do a bit of reading to see if the glasses really work. And guess what? Those silly glasses are not a made-up movie thing. They are actually a real deal kind of thing. The concept is called color blocking. Red lenses allow redding to be visible, but block greening. Green lenses pass greening and block redding. Blue lenses pass bluing and block yellow. This means that drawings in each of these colors become visible when viewed through the respective color blocking filter. The colored lenses of our Ben Franklin glasses allow us to get creative, searching for and seeing a full spectrum of solutions, giving us power to navigate the things we once thought impossible. Think about today. Your community has navigated its way towards its first in-person conference and a natural history study to advance the science. And all of you, or many of you, navigated your way here with children and all of the things that that contains to get here. Tomorrow you'll go home with new information, probably a stronger sense of community. I'm sure, I know I feel it after being here. And my guess is that many of you will move into the next phase of the cycle, the moving forward phase. This is the time when life feels good. You all know this, right? We have clarity and we have confidence in the decisions we've made. And unfortunately, the moving forward phase, the research revealed, is usually the shortest phase for all of us. And so something kicks us back into orienting again. And it doesn't mean when we're in the moving forward phase that we will not have bumps along the way, but rather we're better able to see the direction we're going. For me, moving forward often came when my lenses lined up perfectly and something like a treasure came clearly into focus. I'd like to share one last story with you today that highlights how with time and practice, we rare parents have the capacity to rotate our lenses and find our treasures. Ben was a late walker, not really accomplishing independent steps until he was about 13 months old. My pediatrician assured me that this was in the range of normal, and I should not compare him to Noah, who walked by 8 months, or read anything into it. And since this was before we knew Ben's diagnosis, my husband and I just spent a lot of time doing what every parent does, right? You let your kid walk back and forth between you and you catch them on the other end when they fall. And so finally Ben got walking, and let me tell you, once he got walking, whoa, did he walk. Kids with Sanfilippo have a lot of energy. They go through a period of time where they have a lot of energy. And by the time Ben was 8, he and I could walk 1.5 miles to Starbucks, order a latte and a milk to go, and walk the 1.5 miles back home, all in 40 minutes. I was a lot skinnier back then. Wow. Yeah, we walked a lot. But by 12, Ben, Sanfilippo was a progressive disorder. And by 12, Ben was moving in the opposite direction. His walking was increasingly unstable, and he was always aided by either comforting hands or a gait trainer. But one night, as my husband was cleaning the kitchen from dinner, a rarity, Ben's mind to memory muscle was clicking, and I decided to let him walk around the kitchen without my protective arms around him. And my husband joined us, and together we started to send Ben back and forth between us, slowly increasing the distance between the two of us, just as we had when he was a toddler. And just as Ben sort of made his third loop around our kitchen, and I started to get very, very sad. A big wave of sadness washed over me for all of the things I realized that Ben could no longer do. But there was Ben. He came back to me. He had this big Cheshire grin on his face, telling me that he was still very, very, very proud of his accomplishment. And his smile sent my Ben Franklin glasses shifting, revealing to me a powerful secret. I'm gonna share it with you now. Here it is, ready? At its core, parenting a child with a rare disease is simply that, parenting. Parenting our rare children has more challenges to navigate, but instinctually and intrinsically, the goals for our children are no different. That night, I was simply challenging Ben to be the best he could be, whatever that looked like. And this clarity of my vision as a parent, that I have been and always will be the best parent to and for Ben, was for me like finding the Templar treasure. It was that clarity of vision that gave me the courage to say goodbye when Ben needed to let go, and the tenacity to stand at a RAC meeting advocating to move gene therapy to a clinical trial just 1 month later. And even now, it gives me the commitment to reflect his determination in my work at Courageous Parents Network. I want to return here for a moment to Noah's lesson on lenses. All right, remember how he said that if you added that third lens to the light blocking perpendicular pair, things suddenly got brighter? Bell's theorem. Scientifically, I really don't understand it, and I don't really want to, but he does, so it's okay. But as a rare mom, it really feels right. Perhaps our exceptional children are the third lens. They bring so much brilliance and focus into our lives. They grant us the privilege of deepness. They help us see that when we live at the edge of the deepest beauty and the deepest pain, then everything, our hearts, our world, our community expands. Their strength calls us to our highest capacity and provides us with a dose of courage to do things we might otherwise not have known ourselves capable of. Their determination shows us that even a small victory makes a good day, and that something as simple as a smile can inspire. Their willingness to learn, to try, keeps us present in the moment and shows us that it's not really about the destination, it's about the journey. Well, maybe these ramblings are nothing more than a 50-year-old's battle with her readers, but I don't think so, because you know what? I'm standing up here and I'm looking at all of you, and I can see a lot of Ben Franklin glasses on your heads. Some of them are well worn, they're quick to know the right combinations. Others are stiff and new, still knowing how to manipulate the lenses. But I know that once you get the hang of them, you're going to be better able to orient, more skilled at navigating, and move forward to find your own treasures. That's me. Thank you.
Hello, everyone. I am Natasha King, and this is my husband, Tony King. We also answer to our more common name, Tony's mom, Tony's dad. Tony is our 16-year-old child with CTNNB1, and Jackson is our 9-year-old, and we are from Delaware. Arrow.
The arrow on the keypad.
I know, I'm trying to.
I've never done that before.
There we go. So, gonna take a little trip down memory lane. My husband and I, we met in college. And I had told everyone in my family I was never getting married and I was never having children, and look at me now. So Tony was our firstborn, and he was born on Father's Day, 2007, making it impossible for me ever to top that gift for Father's Day. He was 6 weeks early. Even right from the beginning, they were saying, oh, you know, he was 3 pounds, 14 ounces, because he was growth restricted in utero. And they were saying, oh, he's going to be here for so long, it's going to take a while. But he was strong, he was small, he was mighty, and we were out of there in less than a month. So I think 21 days exactly. Oh, I keep pressing. There we go. So, developmentally, because he was so small, because he was premature, he was sort of going along and meeting things within the window of where he should be had he been born when he was supposed to be born, but you know, which takes longer for preemies. And we didn't really notice anything until about 9 months old, when Tony should have been getting himself into the seated position and he couldn't do it. If we sat him down, he could sit there, but he couldn't figure out the mechanics of getting there. There's also one night later on where Tony taught himself how to pull the standing in his crib, and then he couldn't figure out how to sit back down. So we spent all night going back in his crib and putting him back down. And we go lay down and he would get himself back up and then cry because he couldn't figure out how to get back down. So we're like, something doesn't seem right here. So we kind of went back to the pediatrician. They're like, oh, well, maybe because he's a preemie, this is happening. So let's just get him in some therapy. So Delaware has a great birth to 3 program. We got him in it. And so we started PT and OT. And then I also, because I am a research scientist on Google, started looking at alternate therapies that we can get him into.
And so initially, when we went to go talk to his neurologist, they told us a very basic statement that has stayed with us our entire lives. They told us that Tony would never walk and he would never talk. Of course, when we got that news, it was devastating to us. We did not know why this was happening. We didn't understand it. We were new parents. I knew nothing about children at the time. And so it was very devastating for us. And so about 2008, about 16 months into life, my wife brought me this information about this new therapy, this new physical therapy. It's called ABM therapy. It's the Annette Benil method. And it deals with a lot of neuroplasticity, which essentially is anytime there's an injury to the brain or spinal column, that the body can naturally make reconnections within the brain. And so I was skeptical at the time, but my wife was very persistent about it. And so we traveled. We were living in St. Louis at the time, so we traveled about an hour away to get this therapy. And I have to say, after his therapy sessions, it was almost like we were like kids at Christmas, because after his therapy sessions, about 3 days later, we start to notice Tony making certain jumps in development. It was market jumps. So before he couldn't get himself to sitting, after a few sessions, he was able to get himself to sitting. And so for his first 3 years of life or so, when we were living in St. Louis, we would go to ABM therapy once a month, trudge all the way up to St. Charles. If anybody's familiar with that area, but we'd go up to St. Charles and get this ABM therapy. In 2009, we finally saw an orthopedist. The neurologist that we had saw said that, you know, he's showing signs of cerebral palsy, but then we got a conflicting diagnosis from his orthopedist. So orthopedist said that, hey, I deal with kids with cerebral palsy. I deal with hundreds of kids with cerebral palsy throughout my career. This is not cerebral palsy. I don't know what it is. I can't tell you what it is, but it's not cerebral palsy. So we were given AFOs. Tony was in a reverse walker. Some of you guys have used reverse walkers before. are still using now. And we continued our early intervention, speech, OT, physical therapy. Everybody knows the main therapies that we've all done. We returned to the neurologist probably about 6 to 8 months later, sometime in that time frame. And he saw Tony and he said, well, maybe he will walk. I'm not sure if he will talk. I don't know what's going on here. And so at that point, I kind of lost a little faith in doctors, but that's neither here nor there. But at about 25 months, Tony made his first steps. I was, of course, out of town, so he waited until I went out of town. His grandmother was offering him a piece of candy at the time, and so he made his first steps, and my wife sent it to me in video, and my heart melted that night. Yes, and so Tony does a lot of things for food. So, in 2010, we moved to the great state of Delaware. Part of the reason my job transferred me to Philadelphia. We moved to Delaware because they have a great children's hospital there, the Morris Children's Hospital, and they also are close to CHOP, which is Children's Hospital of Philadelphia, and they have a great genetics and metabolic department there. In 2012, again, my wife approached me about a new doctor, Dr. Paul Jordan in New York. And he was a certified pediatric podiatrist. He saw mainly patients, child patients who had cerebral palsy. And he had his own thoughts and his own theories on how to, and on AFOs and how to treat our children. You're up.
So, yeah, so from the very beginning, I was the person, I was like, I see this child every day. I know that he is very smart. He is very bright, and people will be like, no, there's not much going on. And I was like, well, you're wrong, and I'm going to prove you wrong. So once again, the warrior on the keyboard, I figured out some things, and Tony, from a very young age, saying like a year old, we know that he loved letters, he loved numbers, he loved logos. And he would just look at them and point to them. And I found this program, and this was like, it came on like a little CD, and they had all these cards and booklets. It's called My Baby Can Read. And I was like, I'm going to teach Tony how to read. And my husband looked at me like, you lost your mind. This is like $100 and something. Are you sure? And I'm like, we're doing it. So Tony has been doing this program for like maybe 2 to 3 weeks. And this is, he's 2 years old. So a little video I'll play him. Very good, Tony. Which one said nose? Nose. Nose. Good. Very good, Tony. Which one said click? Yay. So we would do that with all sorts of words that they had in the video. We would make up new cards, and we would just like watch Tony, like just mind just blossom. He loved it. And so from that point on, I am not, well, I am now, but like I don't consider myself an educator, but like my whole family was full of educators. And I was like, well, we are going to figure out what needs to be done to get Tony educated properly. So we were very insistent. He went to daycare, neurotypical peers, preschool. He was in a mixed program with neurotypical peers, and he started kindergarten. We already had an IEP in place for him. I was asking for a one-on-one pair in a typical classroom, and they're like, oh, let's just see how he does. The first day was a disaster, and I was hot. I went home, I was banging on my keyboard, I pinned a letter with the principal, the director of special education, a few school board members. By 7 o'clock that morning, my phone was ringing. It was the principal, he was like, we cannot accommodate Tony at this school, but we have already at a different school that's not too far from us. Tony can start today with a one-on-one para in a typical classroom, and they will be able to accommodate Tony. And we showed up that morning. Tony had a 6-foot-tall para, and we called him Tony's bodyguard because he was like, nothing is gonna happen to Tony. So, as Cliff was saying earlier, that was kind of like the start of the grow of the family, the people that you know that you can trust with your children. And so that was like a really good feeling. He did a couple of grades with his one-on-one para in a typical classroom, and the next said, well, we have this other program that's called CASA, which is communication and speech language. And so basically they have a smaller group of students who have some sort of communication issues, and they give them like their main classes in a small group, and then they push into a larger classroom for other needs. And so we went with that. And what we quickly learned was that the phrase that I coined was Tony rises to the lowest common denominator. So if you don't ask that much for him, he's going to give you even less than that. And if you expect a lot from him, he's going to give you what you ask for. And so we're like, this program is not really working. I would meet with the teachers and they would say, oh, well, we just don't think Tony has the capacity to learn this. We're trying, but we don't think he's going to get it. And so we did that for third, the beginning of fourth, and when we went in December for his parent-teacher meeting, they were telling me the same thing, because they're like, his reading level has stalled, and I was like, yeah, well, you know, Tony's been reading for a very long time, I know he can do this. They're like, well, we don't know if he's gonna get it, and I said, well, thank you for your time, I'm gonna be pulling Tony out of school, and I'm gonna homeschool him now. So I homeschooled Tony, we repeated fourth grade, we got back all the information that he missed. I homeschooled him for fifth grade, and then I found the perfect opportunity for him, which was a middle and high school, small charter school. They had the capacity to give him a one-on-one para and to put him in with neurotypical peers, and Tony went into that environment and he has absolutely flourished. Okay.
So around 2017, while we were dealing with all of Tony's education and some of his physical needs, we also noticed that there were a lot of emotional differences in our child. A lot of over emotional crying, fussiness that were not age typical. We also noticed that he had insomnia. I know a lot of you have dealt with that. Also a propensity to, when he got really tired, he would start giggling uncontrollably, laughing uncontrollably. And I had no understanding of this, but one thing I did notice, what we noticed together was that sometimes these things would flare up after he had certain things in his diet, after he would eat certain things. If he had, if we gave him his multivitamin that had a lot of B vitamins, and we noticed that he would be a giggling mess about 10 minutes later. We also noticed that his insomnia was worse. And so we went to go see an integrative medicine doctor, and they did some tests on his diet and see how he was reacting to certain foods. And it came back that he was reacting poorly to gluten and dairy products and the proteins in dairy. And so we put him initially on a very restrictive diet. It's very tough starting out. A lot, no eggs, no dairy. And this is back when there was not like all the products we have now, the gluten-free products, that, you know, gluten-free cupcakes and things like that. So it was tough at first. But it got to the point where even Tony kind of noticed that when he had certain food products that he didn't feel quite right. And so even he would go with me to the grocery store, we'd go to Costco, go to the grocery store, and he'd start flipping the packages over. Did I eat this? Did I eat this? Oh, I can't have this. And then he would put it back. And so I found that was very interesting. We did notice a very good reduction in the giggling, uncontrolled laughter. Some of his insomnia would subside, but most of all, the emotional outbursts kind of really started to become less and less. So we would have these crying fits for, you know, an hour long, hour and a half long. And those were reduced to more of like 3 to 5 minutes, more typical of a kid going through a temper tantrum. And so we kept him on that diet. He's still on that diet today. We did add some other things, we kind of experimented back and forth. In 2017, partly through my own frustration, we're still kind of digging, trying. Keep in mind, we still don't have a diagnosis. We don't know anything about CTNNB1. We don't know about any genetic cause to what his condition is. So his neurologist suggested another MRI. So we had another MRI done. Luckily, the MRI came back clear, but through the due diligence of the MRI technician, they discovered that Tony's C1, where your C1 and your C2 fit together, his C1 had been broken. And so Tony had been walking around with a broken neck for a very long time, and we had no idea. It could have been catastrophic if he was in a car accident or just outside playing and he hit his head the wrong way. And so the condition is called odeintonium. I hope I pronounced that correctly. But essentially, the only treatment for that is a fusion of the C1 and C2 vertebrae, and that's what we had in 2018. We immediately got his C1 and C2 fused. Also in 2018, again, was kind of frustrated trying to figure out, hey, what is going on with our son? We have no idea. Still, all these years later, we still have no idea. And so it was suggested we go to CHOP, we go to Children's Hospital of Philadelphia, see the metabolics department, because we thought, okay, he's having these food reactions, maybe it's something metabolic. And so they said, no, we don't think that's it, but you should probably get genetic testing done. I said, well. We already had genetic testing done. They said, no, you had screening testing done. We want you to get full exome genomic testing done. Everybody has just probably been through that conversation. And so we had that done. They said you're probably not going to we're probably not going to come up with anything, but we'll call you in 3 or 4 months, see, and we'll have another follow-up. About 3 months later, I was at work and I got a call from CHOP and they said, hey, not for nothing, but we have something. We want you and your family to come in. So we came in, and that day we found out that Tony had a genetic disorder called CTNNB1 syndrome. We went through the same thing everybody else is going through. Okay, what is this? Is there treatment? How many people have this? They said it's rare. And so at that point, there was a sense of relief for us because Tony was 12 years old at that point. And so we finally knew something. And we finally had an answer. About 2019, we got the genetic testing. We also, his doctor, Dr. Jordan, suggested that, hey, he's old enough now to have SPML surgery. And that's with Dr. Nuzzo in Summit, New Jersey, which is, I think, 10 minutes from here. And so he had the SPML surgery. That's a cord lengthening type surgery that a lot of kids with cerebral palsy have. And it was effective. In 2020, some of you have seen this picture before of him with the lined up braces. In 2020, we were officially out of braces. And I've been very happy about that ever since. All right. And so while sound off? Yeah. And so while my wife was working with education and working with developing Tony's mind, I worked on the one thing that I knew I could work on with Tony and I could connect with Tony with, and that's with developing his body. So I 100% believe in what Dr. Jacobs said, that the more our kids are active, the more they move around, the more they use these muscles, then the more it helps them. I noticed very early on that Tony could improve, make measurable improvements in particular exercises and different things that we were doing. And so still to this day, we work out together 4 to 5 times a week. And he has a lot of fun with it. Sometimes he fusses at me, and then he complains and he says, Dad, he's a teenager now. So Dad, I just want to relax. But even he, I think, feels at a certain point, if we go a long time without him moving and keeping himself moving, he starts to, have ill effects or no feelings about that.
So on this slide, I'm just gonna tell you the Mr. Freshman story. So Tony is still at his school. He completed 9th grade this past year, and he got a new para. It was his first male para since kindergarten. And when we met Mr. Johnson initially, him and Tony clicked instantly. They were just so in love with each other. He was his biggest fan. And so homecoming time comes up and Mr. Johnson shoots us a message and he said, Tony says he's going to run for Mr. Freshman and we 100% believe that he can win. And I was like, okay. And he's like, Tony is the mayor around this school. Everybody loves Tony. We're definitely going to win. We're going to campaign. And we were like, all right. So from the very beginning, I would ask Tony, I'm like, you sure you want to do this? And he's like, yes. And I'm like, do you think you're going to win? And he's like, of course I am. So like, he just never doubted it. And for me, that was so reassuring because there's so many people along the way that was like, your child's not going to be able to do this. Your child can't do this. And so when you meet someone that's like, no, your child can, and we will, it is phenomenal. And so I chaperoned homecoming, I was there, and when they announced that Tony won Mr. Freshman, he went, yes. But it was like he knew it all along, and his parent was over there crying, and Tony was just like, yeah, I knew. So my words of encouragement to everyone is that you are your child's biggest advocate. You have the Ph.D. in your child. You are there with them all the time. You know more about them than anyone else ever will. So don't let anyone tell you that your child can't do something. They may have to do it differently, but if they want to do it, and I've said this to a bunch of people here, these kids are sassy. And so if they want to do something, they're going to figure out how to do it.
Yeah, thank you.
That was fantastic. So we have one more, Jennifer Steinway's gonna come back up. I think she's on here and do a little workshop with us. And then Emily will be just doing some quick closing remarks.
Thank you. So I'm gonna be a little less formal this time. And I'm gonna start by saying, what I really wanna know is to hear about what you guys are having difficulty coping with. I can give you a little didactic learning up here, which might help you, but the real deal is that I just wanna be a resource to you, not just as a rare disease parent, but also as someone who works at Courageous Parents Network and has the opportunity to interface with a lot of families around coping issues. The real reality about caregiver coping, and it's not my favorite topic, to be honest with you, because the real reality is what we need to cope is more. We need more help. We need more resources. We need more time. The real list of mores is huge and big and long, and I can't solve those for you, and they're not solvable in some ways, and that's just the real reality. I will say that, like Natasha said, when you know that you're the expert on your child, I think a big part of coping comes from really absorbing that and really embracing it and know that you are the expert and you are doing the very best that you can with what you have in the moment. And I know that's really hard to know. I mean, my son Ben went through a phase where he was only sleeping 3 hours at night. I thought I was gonna not do well. And, I ate a lot of cheeseburgers from the drive-through at McDonald's because it was the only time I had to eat because I had to sleep when my other two children, so that I could parent my other two children. So I can go into this lesson, but I'm also happy if you guys want to just ask me a question, if that would be more productive, I'm happy to do that. So just interrupt me, okay? Because really, Effie knows this. I'm better off when I just riff. Let's see. Let's see, maybe if I press this. I got it. Oh, it's very responsive, this little board here, huh? Okay, so a lot of times when we talk about, we're going to talk about like the real stuff now. So just again, interrupt. When we talk about that feeling of not being able to cope as a caregiver, We commonly call that a feeling of overwhelm, okay? It's an actual real deal. You can suffer from it, you can read about it online, and you can find some strategies out to help you, which I'm hoping some of these today will help you when you're feeling that way. And we don't always feel that way as caregivers. You know, in my little talk earlier, you heard about that cycle of orienting, navigating, and moving forward. Those are happy terms for like the real scientific parts behind it. But overwhelm often happens during doing orienting, right? Something new has happened and you're trying to get used to something that's different and unexpected usually, or challenging. And this is when coping can get really hard. So I want to just give you 5 things that you can try. I want to keep this short because the day's getting long. It's the end of the day. But these are 5 simple things that if you remember when you're feeling those feelings of I can't cope and I don't know what to do, or I'm feeling overwhelmed. And of course, I would also just suggest, don't be afraid to embrace a professional in that process, and certainly lean on one another. But these are 5 easy things that you can lean on. One is recognizing triggers, and I'm not gonna read this list actually, let's just keep going. So it's really important to recognize the triggers that start to cause you feeling like you can't, you're feeling overwhelmed and you're not coping well. A change in baseline is one that I already threw out there. These are triggers that are the most identified triggers from the families that we polled at Courageous Parents Network, okay? So that would be a change in your child's baseline. If that's not a word you're familiar with, it's a really handy one to have. That's when some difference happens in your child. So in the case of Ben, in my story, it might have been when he went from being a good walker to starting to stumble. That would be a shift downward in baseline. Kids' baseline can shift upwards too. So it's not all negative, but even the upwards shifts sometimes present new challenges, right? Your suddenly immobile child became mobile and you have to rearrange your house, or they've learned to master their wheelchair on their own and you have to accomplish something. So, you know, to modify for that. So those can be a trigger. Changes in plans or times of transition. This is common. You already all know this, right? I just heard a lot of stories about this. This could be your child switches a school, any of those things, but also recognize that it can be something as simple as the babysitter canceled. Okay, and it often is just because the babysitter canceled and you're just needing to go out and do something like the person that you used to be. A different and odd one is exposure to information, but remarkably we hear, you know, the internet is a blessing and a burden. So pay attention and make sure that what you're looking at, if that's Facebook and you're seeing complications in a different family and it's triggering you to think the complication is coming for yourself, you're reading about something online. I know it sounds silly, but it can be a really complicating trigger if you're feeling a sense of not being able to cope. This one is obvious, and when we all do it, you know, we all long for that normal life, the life that we thought we were supposed to have before the diagnosis of our child. We can't help but do it. You know, you're out in the world paying attention. We all do it about other things too. Just know that it's normal and figure out when it's happening and if it's happening to you. And this one is kind of surprising, but unwanted advice can be a trigger often to make you feel like you're not coping as a caregiver, because what it often does is it makes you feel like you're not the authority. When somebody says to you, they give you this advice, it's like you're not doing, the self-talk inside your head starts to say, am I not doing this right? Am I not providing the right way? So pay attention to the advice you're getting, and if that's a trigger. You're going to get your own list, but I really encourage you to sit down and think about a period where you were trying to orient to something new, or you were feeling like you couldn't cope, and just identify your triggers. So at CPN, we have a motto. We say you have to name it to tame it. And we talk about this a lot. It's actually a pretty good strategy that's used in a lot of therapy sessions. And so part of moving out of the orienting phase or that feeling of coping is to identify your feelings. Too often we say, I'm fine, it's okay, it's all good. We don't really talk about our feelings. And I think that some of the reasons why we do that is because we tend to be problem solvers, and so we push away the feeling and go straight to the solution. But naming your feelings can be a good strategy to helping you cope. One thing my husband and I did, and we didn't do it as formal as this little exercise that I have up here, but it really works, is I'm a really stressed out person. Like I go right full tilt. And Effie knows this about my husband. He's pretty mellow. He's like, never. And so often I'm the one that ramps up the caregiver, like not coping thing, right? I'm the one that gets in the cycle. And we finally got in a strategy where he would say to me, are you looking for a solution here or just acknowledgement of how you feel? And it was very enlightening to me because I thought I was driving towards a solution when I was escalating through those things, but what I was really looking for was acknowledgement for the way I was feeling. And acknowledgement goes a long way. I mean, validation is so affirming as a person. And, we just sat here and talked about all this scientific stuff, but really a clinical trial is just trying to validate something. It's in our human nature to need validation. And so try to check in with yourself and your emotions. Don't just run to the answer, because somewhere in there, the emotions are going to come back and they're going to influence your caregiver coping. This one's really big. It got 3 slides. I'm going to go quickly through them, but focus on your strengths. When you're not coping, it's really good to know what your strengths already were. They are your superpowers. They are the thing that's going to help you pull out of it. Oops. Sorry, I'm having trouble advancing. What? Don't get up. You're pregnant. Don't get up. Let me go backwards, and then maybe I can go forwards. Okay. There we go. Okay. So honestly, I'm going to be truthful. If someone had said to me when I was caring for Ben, make a list of your strengths, I would have been like, what? How the hell am I going to do that? I don't have no idea. So I made you guys a little cheat sheet. I know they're going to send you around this. But these are some of the things you can do. Think about a time in your life when you were your best self, okay? Like the place where you were most happy and you thought you were doing a good job. That could have been 10 years ago. It could have been yesterday. But think about what you were doing and what were the strengths that you brought to that situation. Try to name some positives about yourself. This is really hard. Most of us don't like to do that. It feels awkward. So ask somebody else. They're probably pretty good at identifying your strengths. What are you doing when you feel the most confident and feel the most relaxed? The relaxed one is really big for me. Like this is when I discovered that I feel the most relaxed when I'm cooking. I know that sounds very odd, but we're a big cooking family in our house. And part of it is I realized that the mundaneness of chopping let my brain just sort of let go and solve. And so by understanding that strength, that I could go to a mundane rote activity when I was feeling stressed out, like chopping, was a big awakening for me. And also look at some of the things that you're good at, and then you can do the opposite. I'm the kind of person that usually looks at the negative to find the positive. So sometimes looking at what you avoid can help you to identify your strength, because you can say to yourself, why am I avoiding that task? Is it because I don't like it? Is it because I'm not good at it? All of these things can help you strengthen your ability to cope. So I'm going to encourage you to do something nerdy like this. Make yourself a little chart. Put it on sticky notes so you can remind yourself. It's helpful. Also, you can use it to say to somebody, I don't know what to do right now in this situation. Look at this list of strengths I think I have. Show me which one you think I should use right now. Sometimes you just need somebody to tell you the answer, right? This is my next one. I like to say in rare disease, when our child gets a diagnosis, we are cast into 2 roles. We are both the driver and the passenger. Because so much of what we have to do is about decision making, and decision making is really hard. And it's fatiguing. There's actually a thing out there called decisional fatigue. And we are chronic decision makers. And we are forced to be in the driver's seat, but in many ways, the experience we're having parenting our rare disease children feels like we are passengers and that we don't have control. So when you start to feel like you can't cope, just sit back and say, am I feeling like a driver right now or a passenger? Because that can help you also to identify what strengths do you need to bring to the table, or what seatbelt do you need to help you, right? Because you can reach out to somebody else. So thinking about your job as parenting as those two roles can help, and try to focus on things that make you feel like the driver. Okay, you all know this because you're here, right? You know that finding community is super, super, super important. There's nobody that parents listen to more than other parents. This is the entire platform of CPN, is parents talking so that you guys can all get resources from other parents, the most trusted resource. So finding your community and doing all of this is really important. But I also want to stress to you that when you're feeling like you can't cope, part of your community also means reconnecting with the person that you, I don't want to say were before your child's diagnosis, but the person that is internal to you, and making sure that you also have people in your community that relate to that person, because you still are that person inside and you need that person tended, just like you need all of these other things that you're tending over here with your disease community family. So don't forget to reach out and find a sense of belonging, because it really can help wherever you find it. And then I would say to you, another great resource is Courageous Parents Network, which is just an online resource. You can go on, you can watch, you know, all the videos. We have 700 video segments of parents talking about challenges that they faced. Some of the things probably won't be relevant to you, but some of them will. We focus a lot on decision-making and medical care, transitioning our children to adulthood. You're going to find a whole lot of resources on there. We have an app if you want to do it on your telephone. That's not me. I'm 57. I don't really do anything good on my telephone. I got good at taking pictures, but that's about it. Oh, I went the wrong way. Oh, I'm still going the wrong way. Anyway, check out Courageous Parents Network. I really encourage you to. And also, I'm just a rare disease mom trying to do good work for all of you and all of the other moms and dads that are out there still doing this, and the siblings. I mean, the siblings are such a big thing. And, you know, I got 2 of my own that are still making their way, even though they're in their 20s. But this is my email address, and please don't hesitate to reach out if there's anything we can do. And now what I want to say, is there anything that you guys want to ask a question about, like coping wise or rare wise or any wise? I'm happy to answer. I know you've had a long day, so you're probably just looking forward to, yeah, getting a glass of wine, right? All right, thank you for your time and your enthusiasm, and even more, I guess, also thanks for everybody that shared their kid with me today, because that's my favorite part about coming to conferences, is I get to check back in with my kiddo love. So thank you.
Hey everyone, I'm Emily Amerson. I joined the board a few months ago, got my daughter's diagnosis a year ago. So I'm just gonna close us up here. Thank you all for being here. I'm just gonna talk really quick about getting involved too. Before we all leave, sorry, I know everyone is so tired. And Jen stole my analogy, Mojo, but I just wanted to say, the car is there, the plane is there, we're the gas. You know, we know where we're trying to get, but we need all of us to get there. So it's really important that we are involved as much as we can, and that might look like a certain season, maybe 6 months, maybe a year, maybe 4 years. You know, just as much as you can put into this, it's gonna push us towards that end goal that we all want. So if you would like to get more involved, our website has a volunteer form. I would encourage you to reach out through that or any of our board members. And we'll get you plugged in. We would love to get you plugged in. Obviously, thank you for being here. That's a huge step. And we just want to keep moving towards our destination. So please follow us. Please stay connected. The natural history study, it's not a one-time thing. So everyone who participated in that today, yesterday, Thursday, please stay connected because that is what's going to be meaningful for that research. All right, thank you guys.